Megan Wallstetter. I've been working to put on these Growers Association, Cannabis Association events. This is our third event this year. We've had a happy hour and a legislative update, and we'll announce a fairly big event at the end of this event, so you can save the date. We kind of consider ourselves a little association that could, because we just keep going. It seems like against all odds. So we are excited you are all here. We actually have 100 people here, which is awesome. And part of what Megan and I and Charles, who's been here, and there are other members here in the audience, have been working on is doing something I think that's a little, for those of you who don't know me, I'm a lawyer, something a little bit different, which is trying to provide some like practical skills workshops to talk about things other than just kind of the ongoing legislative updates, ongoing legal talks. I mean, those are important and we'll keep doing those, but we also want you to be better cultivators and better processors and better participants in this industry. So we reached out to Jeremy from FARMA and Jeremy from Cascadia. I don't know if any of you heard either of them talk, or maybe you get your cannabis tested with them, but I've had the opportunity to hear both of them speak a number of times. Jeremy is from Cascadia Labs. They are a great lab. They recently came or expanded over to Bend to here, although they've been serving all of Oregon for some time. They are quite fabulous. And Jeremy from Pharma is really great. But if you can't hear him, you should let him know because we don't have a mic. So they are going to talk about some of the in-depth science work that they've been doing. They're going to talk about testing. They're going to talk about tissue culture. And I'm hoping that you guys really enjoy it. I think what we'll do is we'll have them talk and we'll maybe go until about 6:15, 6:30, and we have food coming. And we'll do a little bit of a happy hour. And you can ask questions, you can talk, and we'll also announce a new event. So Jeremy and Jeremy are here, and feel free if you can ask questions. This is meant to be a learning opportunity and not just kind of a talking head, so to speak. So please give them a round of applause. Thank you.
Thank you so much for being present. It's really exciting to be here with you and to actually get into some more depth than typically is being served in all of the popular media. There is some just crackling stories emerging in our movement that are really being under-attended, and it's our joy to actually deliver some of that content. There's been a lot of research going on and a lot of discovery, and it's just a time of great insight now that science has come into the field.
That's correct. Yeah, so I'm sure anybody that's been participating in this industry over the last year or so has really seen evolution of obviously the regulatory environment as well as the testing that is going on here in Oregon. And one of the phenomenal pieces of one of the greatest points of that is Oregon is a leader right now across the board, across the entire United States. Both with what we have within our medical system, as what's coming through with the adult use programs, obviously, but also from just a fundamental level, the folks that are operating here, both the growers, the extractors, the analytical labs, the research labs, we have something really special here, and it's really exciting to be a part of. And I just wanted to thank Amy and the Growers Pack for inviting us here to chat today, and all of you for coming out. I personally think that Mr. Plum here is going to have a great start with a little bit of kind of the history of cannabis, some of the genetics, where we got to where we are potentially, and some of those stories that you can speak on. I'd like to really speak on some of what we're doing just from an analytical standpoint in the chemistry microbiology lab, what we're looking at currently, what we see coming down the pipe with future regulation, and kind of where that's going to standardize the lab practices to better serve the industry and the communities that we're all a part of. So that's kind of where we're coming from, I guess.
And maybe, I wonder if I could just do a small block and then maybe
So I want, before you get started, we're going to turn the AV on so you can all hear, so it might be super loud in 2 seconds.
Okay.
And I may need Claudia to speak louder. It's not my style. So, as Jeremy's enunciating, and I want to continue with that, I really think it's an amazing moment to celebrate the resources that are unique to our region. But I've been involved with cannabis my whole life, and I travel a lot, and I've been many places where cannabis culture has flourished. But what I recognize clearly, and I think more than even a lot of people who are here in the midst of what's happening, is that there's a really unique constellation of resources here. And that the fact that we've had like 15 years of sort of de facto decriminalization with the letter Tom Powell submitted a long time ago saying this was the lowest stated priority, cannabis-related issues in terms of persecuting all the offenders of violators of cannabis laws. The truth is that for 15 years, there's been a community here with tremendous quantities of growers, sort of orbiting grow shops and having access to resources that, like, if you were in Northern California, you would not have had that same density of people interacting in hubs with scientific resources, and a real emphasis towards breeding and creating an incredible diversity of genotypes. You know, we are blessed with one of the greatest inheritances and heritage of essentially the most vast genetic diversity of cannabis anywhere on the planet. And really, I mean Oregon. I believe that one of the dynamics that's been involved in Northern California's production is that there's even more of an emphasis long-term on epic-scale commercial production and reducing genetic diversity in a sense. There are amazing breeders throughout Cali and everywhere else, but Oregon has a very special piece where there's been a lot of small, isolated, and sort of more urban, different groups of growers working passionately without the intention of producing OG Kush, Blue Dream, and Sour Diesel exclusively for these coasts. And there has been this incredible high usage rate in Oregon, and that there's just a lot of things that we're fomenting for a long time. That what's true, like mycelium and so many things that are happening quietly beneath the soils, things emerge sort of as if from nowhere, but there's been an incredible networking happening below the soil. So I do want to speak about a little bit of the reframe that's happening, because one of the issues we have in cannabis, and as we're becoming this massive industry and having a lot more influence in mainstream culture, is that it's time to revisit a lot of the basic ideas. We take for granted a lot of the taxonomy and categorization that we put the plants in, and that we have come out of a black market era where we just didn't have a lot of insight, like the insight any horticultural field would garner if it was gathering botanists and molecular biologists and phylogenetics efforts. I mean, really, cannabis is just suddenly having this light of tremendous resources showing up. And it's truly amazing what we're finding. But one of the things I want to do here is we have a pretty elite group. Anybody who's present tonight is in their own edge in relationship to the industry. And that I want to start with a reframe of fundamental ideas that then leads into a reframe of kind of the creation mythology of, like, how did cannabis come back into humans' hands after the Pleistocene? It's a tiny piece there, and then I want to give just a few technical, exciting bits about the research that's cutting edge and some of the insight that's coming through that, and then give it to Jeremy for more analytics. But so starting out, I really want to address the fact that when we're talking about genetics in cannabis, one of the key issues that's a big deal is that we almost have no means of communicating with each other in a fashion that has a baseline. What I mean is that if I have 15 skunk number 1 seeds from 1991 and I crack those 15 different genotypes, each one of those is not the exact same thing. They are a different genotype. But in our industry, what's happened in the black market is we've started to call strains or phenotypes what is actually a genotype. So I want to explain the difference here because it will help us to actually speak at greater depth about how to work with the plant. And so really, a genotype is the DNA. When you start a seed, that's a novel expression of an XX-XY pairing. That's never been seen on the planet before. This is evolution in action. But the genotype goes into an environment, and like nature and nurture issues for humans, with plant genetics, it's amazing. I mean, we have 10,000 base pairs of DNA as a human organism. that actually a plant will vary between 50 and 150,000 base pair of DNA. It used to be the case before we've done whole genome sequencing very recently, that we would conceptualize of the most evolved organisms by the amount of genetic material. So just at root, we are reevaluating our position in the evolutionary schema if we look at the fact that the plants have incredible genetic diversity and can adapt in ways we can't even conceive of yet. That there's enough information genetically to respond to environmental triggers and influences that are far beyond what it has ever experienced. It's set up for unfathomable complexity and depth of adaptation. This is the truth about the plant, and it's a dynamic, fluid, flowing process that's really, right now, at an incredible point of discovery, and we're finding a lot more than THC and CBD. But the genotype must be distinguished from the phenotype. The phenotype is when you take a genotype and then it interacts with the environment. And then the environment interacts with the interactions of the environment. But for simplicity, it's simply that you've got a genotype that now is being produced with different fertilizer, different lighting, different levels of CO2, different levels of all sorts of environmental input and stimuli. This is going to create a novel chemotype. The chemotype, in sort of my work at PhRMA, when I'm serving patients at the dispensary, is the relevant information. What really matters to patients is being able to have some sense of what are the baselines, what are the compounds in this particular flower. And that's not going to have anything to do with the phenotype or the mislabeled genotype, the strain name. And one of the ways I can illustrate this is that I gave out 4 cuts of blackberry kush about 2 years ago. And I gave them to master growers, and they each produced a version of it, and I produced my own version. At the end of it, we tested this for a full panel at Sunrise Analytical, and we looked at the full 45 terpenoids, 22 cannabinoids, 15 kinds of THC, in a really extensive report, ultimately. But what happened was that these flowers looked and smelled much alike. But when actually we looked at the lab data, what was radical is that as a grower, I would have expected there would be a nominal range that the compounds that would exist in that genotype would be expressed in. The fact is, my mind was blown when I saw the results, and all of these amazing growers produced results that were profoundly unique. So in one, a massive myrcene peak, a terpene that is associated with hops and cannabis that is common to both, and is really one that we should pay attention to because it has a very powerful effect. It really leaves about an hour to hour and 15 minutes for THC in the euphoric experience before myrcene biometabolizes, and we know a lot about myrcene from the hops industry, but an hour to an hour and 15 minutes after consumption, myrcene is probably going to go off like a bomb. You're going to be really tired, and you're going to be checked out, and it's going to be an undesirable side effect in a lot of sort of measures. However, it also has profound therapeutic benefit. Myrcene as a compound that actually facilitates and makes more permeable the blood-brain barrier and helps to facilitate the uptake of many other cannabinoids that otherwise would have a challenging time. So there's also an anti-inflammatory and antibacterial properties. Myrcene is not a bad thing, but we should be tracking it because it's one of the more influential things in determining what the effects of a particular phenotype are going to be. Getting back to this really simple idea, is that really, if nothing else happened, we could start to speak at least amongst each other in a way that would have a shared baseline. This would really be a boon to the industry and to all of our collaborations. But that a genotype is a seed, and really that's a novel seed. And then once you brought out this sort of seed into your environment, you have a phenotype of whatever that was called. But the issue is that that strain, which maybe you've got a Chem4, and then there's, of course, Chem4 everywhere. Everybody has different genotypes of Chem4, and this is resulting in a very problematic situation where we're never going to be able to speak to each other about what's going on. So this is what's exciting about what Phylos Bioscience is bringing to the industry. One of the things that we need as a tool, as both producers, as professionals serving patients in dispensaries, as researchers, we actually need to be able to have a shared language and jargon that works and that supports good scientific process. So ultimately, the issue with the phylogenetic work is that they're going to be able to do genotyping sort of certification. And this happens, like if you're producing apples in Washington or in any other horticultural field, it's a big deal that the Fuji apple that the farmer is sowing actually is genetically accurate to the Fuji apple that the consumer expects. And that's been a plant that also was bred with a lot of pest resistance and was a very virile plant that could withstand commercial production, something we're not seeing a whole ton of in cannabis right now. And the reality is, after they genotype, they're looking at it DNA and saying, yes, this is Fuji. But that's what's about to happen for cannabis, and it's going to be a big mess. And a bunch of people are going to be really pissed off. Of course, everybody will claim that they've got the right version, and in a way, everybody does have the Chem 4 that is of yore. It's just that we're about to cross a line in the sand, which is going to say, actually, we have to start with the baseline somewhere. And by the way, it's just happened recently in the world of botany in a much larger sense. I mean, it was literally in the early 2000s when we started this effort towards moving away from Linnaean taxonomy, categorizing things by affinity, to actually looking from an evolutionary perspective at the genetic material and finding out, like, for example, that hops in cannabis 4.5 million years ago were the same plant. So we can say that they're then in the family Cannabisae. But we're now going to go into this new realm where I know that people who get their material into phylos ultimately are going to have at points a contentious process as we start. But I promise you that what is waiting ahead for us, both for what we can do, the good we can do for patients, the utility we can provide as an industry, it's profound. We must start there. It's coming, and I want you all to know that you can expect at some point certified genetics, and that's not in any way going to capture the whole market or be the end of the story. Because one of the big things with genetics is that we now have an issue where we've cloned ourselves into a corner. And what I mean by that is that we're actually reducing genetic diversity in cannabis in a really serious way. And that if we care enough about this plant and about the role it plays in our relationship, that humanity and cannabis has been an ancient bond, then we really have to invest collective resources into a kind of breeding program that restores some of the incredible genetic diversity that has been in cannabis in the past. And what I mean by this, now I'm going to, I'm attempting to kind of blow up the idea of what people are calling strains and phenotypes, and suggest that we start to use genotype for an actual, like a strain, and that a phenotype is your version of that strain, and the chemotype will change every single round. It is the data that a patient needs to see so that they can actually start to have baselines. Last year, he publishes his follow-up, I mean one of many follow-ups, but this is the giant life's work moment, where Robert Pennell Clark, after a lifetime of being involved in this field, writes the definitive account at this juncture of ethnobotany and evolution of cannabis. And I wish I had started at the end, because I would have gotten this key insight way earlier. I've been working with this book for a while. Chapter 11, 12, and 13 is where I want to summarize it briefly. This has a really key insight. So what I mean by cloning ourselves into a corner with genetics is that what we've been calling indica and sativa are sort of figments of our imagination. There is no basis for a taxonomy that actually refers to this plant as an indica, sativa, and a ruderalis. Pennell Clark in Chapter 11 does a thorough review of every attempt to put cannabis into a taxonomy that's meaningful and productive. And so he reviews all of the scholarly works over time that have evaluated the same problem. And there's been a lot of different ways people have tried to categorize it. But I believe his new perspective is the most useful, and what I'm going to argue we should all consider being able to at least speak amongst ourselves when we're talking about cannabis, especially when we're working on breeding or working at a genetic level. And it's that there is narrow-leaf hemp cultivars, narrow-leaf drug cultivars. There's broad-leaf drug cultivars and broad-leaf hemp cultivars. There's also a fifth category, which is putative ancestors. Putative, as in we're reputing them to be there, but one of the issues with ancestors that we've discovered is that one thing about cannabis is it produces huge amounts of pollen. One of the sensitive issues as hemp comes into production in Oregon, pollen can cover the entire state in one corner of the state, is what it would seem like. But the reality is that when a new genetic shows up in an inbred land-raised population, it quickly establishes itself. It hybridizes in a widespread way that actually for all time changes that population. So in one single growing cycle, you could have a strain that was inbreeding, and what I mean is for thousands of years, landlocked in an ecological island, and kind of creating great repetition with very narrow variability, with very small amount of mutations. And that you could take that and add one Dutch genetic to that Malawi plantation, And from that moment, that Dutch genetic is a part of everything there. Those genetics have for all time changed, irrevocably, and for good and bad. But in this case, what I want to say is that if we're thinking of breeding in terms of sativa, indica, and ruderalis, we don't even have a tool set to approach the task at hand, which is actually to restore an incredible amount of genetic diversity that will adapt to changing times. Because we've had climate change before, not this version. This is the Anthropocene. It's human-induced. But really, the last ice age, about 12,000 years ago, marked the end of the Pleistocene era. And at the end of the Pleistocene, there is this warming. But during the Pleistocene, through the ice age, there's these little islands where plants are actually still fertile and remaining either viable in seed form or actually growing in a few places. But two places now we can trace with modern genetics back to their origin. One place is in Eurasia. And I believe this story goes like this. This is Conalpar's version. Essentially, you've got a migrant sort of population, you've got band-level culture, groups of 50 or less that are sort of in a seasonal round. And they're gathering seeds and they're gathering all of the resources and ultimately they're having settlements. So people are gathering cannabis seeds, and the cannabis seeds at the bank of the river turn out to be an oily, delicious, nutritive crop that obviously is a useful thing. As the seeds are collected and brought back to the settlement, then the seeds begin to be sown over time. The very beginning of agriculture commences potentially in a way in line with cannabis and humans, and there's a lot to say there to develop that, but I'll just say that that's one theory, that there is actually an early inception of an agricultural modality with cannabis. But then what happens is eventually these sun-loving plants, they're particularly adapted to the phytochrome receptor that receives far red light, the PFR aspect of the phytochrome. And this is the ability to receive light in a shaded area. So for example, if you're in a forest floor and you're shaded out, and you have this amazing phytochrome that can actually receive the far red light that is not absorbed by plants, while the plants are absorbing the near red, maybe 680 nanometers, the far red, around 730 nanometers, is penetrating the canopy, and it's awakening the seeds. It's awakening the dormant plant that's shaded, and it's signaling this plant what's happening with the PFR levels inside the foliage. Grow fast, be lanky, extend until you find light. And so there's a particular plant growth response as a result of that frequency of light showing up. And this is a little precursor to photobiology, which is what I want to get into and where some of the really exciting stuff is happening right now. But in this case, this is also an evolutionary aspect. These plants that are sun-growing around human settlements start to grow up widely wherever people are. So where people are making settlements, cannabis is there as well. And then we reach out and there's bast fibers that are growing from floor to ceiling that are instantly rope growing from the earth. And now it's clear that actually, like, even when we domesticated horses for the very first time, that was because we had access to hemp fibers. It was the unbelievable convenience of these bast fibers. Looking back to basketry, to all kinds of craft that involves fiber work and textiles, hemp was absolutely central at the very beginning, as well as at many waves of culture every generation since. But the amazing thing is that at some point, humans are moving all around, and they start to go up in altitude. And this is, so the idea is, why cannabinoids? Why secondary metabolites? And really, in general, the answer is almost always pest defense response. Sort of knee-jerk, well, it's because this is how plants protect themselves from predators and pests. That's true in the case of cannabidiol, the evolutionary first cannabinoid. And that actually, the CBD is, in the early, what we were calling landrace sativas, which were the really lanky, narrow leaf hemp cultivars, which were the prehistoric earliest versions of cannabis, were actually never having THC at all. That didn't happen until much later. THC is an evolutionary product that happened as the plant moved up in altitude. As the altitude increased, so did the UVB levels. And this is the consensus position in researchers that I'm seeing right now, is that the UVB levels were damaging enough to the plant cells that eventually these incredible terpene-phenolic compounds, the terpenes and cannabinoids, begin to get more and more complex. CBD, not so great at blocking the damaging effect of the sun. That as the plant's developing more and more, eventually we get to THC. THC, it turns out, is impeccable sunblock. It's the best thing that's ever been evolved in nature to shield against the harmful UVB light. So this incredible catalyst of following people, and unwittingly people participating in the transporting of these genetics that were really purely distributed in an equatorial band around the planet, it looks like. Ultimately, the plant evolves with us and we evolve with it. And of course there's the argument that the THC obviously was very attractive to the people who were in proximity to the plant, and that it was a coevolution with multiple strategic advantages, as is the case in most of the adaptations that succeed. So at some point, we've got now high THC cannabis, and at some point we actually begin to use that for the purpose of the THC, being a therapeutic compound, a spiritual sacrament, and so many other things. Ultimately, this is a new cultivar beginning, and that where we must distinguish between the narrow-leaf hemp cultivars and the narrow-leaf drug cultivars is that they continue to have different purposes over time. There really was an evolutionary parting at this period of time early on, and we still, to this day, have broadleaf hemp cultivars and narrow-leaf hemp cultivars that will produce no THC. And the reality is, there's only one camp that we're not really playing with at all, and it is the most important thing that we can all be working on. And I'm going to be honest, this is like trade secret IP interpreted from the most advanced cutting-edge science. And my wish is for our region to flourish. It's for us all to collectively recognize the unbelievable opportunity we have with the science in our midst and the creativity and this craft ethic that is so Portland, to go to another level of consequence in the industry and demonstrate to the world what's really possible here. But what I want to say is that the broadleaf hemp cultivars, the narrow-leaf drug cultivars, the broadleaf drug cultivars all come from the same genetic source. This is so we're now going to call all of those plants indica. Everything on your dispensary shelf, everything you've probably ever touched and seen, with a few exceptions, are indicas. Cannabis sativa is a family. Or Cannabis is a family, it's a genus. But actually the indicas are referring now to all three of those categories of plants, and the putative ancestors are sort of hovering with no identification. But the narrow-leaf drug cultivars, or excuse me, narrow-leaf hemp cultivars, are in a totally different camp. And this is the significant thing. This is where our work must begin, and has begun in some cases. It's that actually if we go to Northern Europe, as that population in Eurasia was breaking out after the end of the Pleistocene and the Holocene was beginning, then this whole other camp starts to move around and is isolated by huge geographic diversity. There's incredible terrain, and sort of all this topography encourages real isolation. What this results in is a plant that is genetically different than these other plants. And what I mean by that is, we now have a genetic basis for talking about this categorization. There's a BD allele and a BT allele. These are two genes that are basically the most important gene associated with the biosynthesis of THC on one hand, or the biosynthesis of CBD on the other hand. So one of the amazing things is that in the indicas, and what we're now calling indicas, we have always both. Both genes exist, and therefore you can have all the possibilities, which Robert Carnell Clark defined the chemotypes in like late 1950s, and it started this process, but here's the truth. He knew this already way back then, and we still don't know it now in popular culture, that there were three chemotypes. There's a high THC chemotype, a high CBD chemotype, and a 1-to-1 that varies between 7 and 13% either direction, so between a 3-to-1, a 2-to-1, and a 1-to-1. And that those are the, if you cast seeds from all around the world, statistically, you're going to come up with those variables. Over time, you'll figure out there's these three. So you'll never have, for example, a plant that's the epic 1-to-1 on both sides, because there's actually competition for enzymes in the production and synthase of those compounds. But what's really interesting is also, we have had results for a very long time from gas chromatography and serious lab work that showed potency that was 25 to 30%. Long time ago, Carnell Clark and Watson and people and their crew were able to identify those levels of potency. So it's always not true when the media is saying, we bred super cannabis that's so high in THC. And meanwhile, there's the new accusation, slightly coming from a group of friends from LA, that actually we've also, like all of the West Coast growers were so irresponsible because they disallowed CBD into the equation. They actually had CBD to begin with, and then it went away. And so these reckless hippies. And this is not the case. What I'm going to tell you is actually there's a bit of like kind of heroism that we must celebrate in the underground culture that is why we're here able to do what we're going to do now. It's that through incredible outbreeding, which is different than inbreeding. Inbreeding, again, happens in a sort of land race area where for thousands of years these similar genetics are crossing again and again. But what's happened on the West Coast is we just kept finding new things from everywhere around the world. We added everything we could ever find and some things that didn't even fit into the equation, and that's been a stew fomenting for decades. What's happened is I've never seen a potency of THC much more than 30%, or at least that I would trust. But that I do believe that we've actually seen an increase across the board of all other secondary metabolites. Most people don't know this because most people don't get to see the lab results we've seen. One of the things that I'm really going to encourage everybody to lead with is the sense that whenever you have extra money, if you want to learn about cannabis, right now is the most exciting time ever. Give it all to the labs. Literally, and don't hesitate. Know that they're able to shine a light into the dark that nowhere has humanity ever been able to navigate before. But one of the things we've seen at Pharma, and I do believe right now we're sitting on more chemotaxonomy, more chemotype data than anybody on the West Coast outside of Michael Backus's group, Care by Design in Los Angeles. And that ultimately, we're really working to have great insight in a sort of comprehensive way of the kinds of variables that are out there. Of course, we're still limited. We're only tracking and measuring what we can measure. But we're doing it all very well and creating strong data. And one of the things I'll tell you is that in looking at new hybrids, there are extraordinary cutting-edge hybrids that are really popular. If you look at any of the Girl Scout cookie phenotypes, for example, those plants in general are producing huge amounts of caryophyllene, which is, by the way, a truly desirable compound. I'm noticing in my personal use that anytime I find a plant that has an outrageous caryophyllene peak, it's also got anti-anxiolytic properties. It binds weakly to the CB2 receptors, that are the same receptors that cannabidiol binds to, and has some of that effect, but without the dissociative effects that I find in the entourage between cannabidiol and THC, and that I've seen a lot of depressive episodes and really strong issues in my patient work using CBD. Caryophyllene seems to have some just amazing therapeutic attributes, but this anti-anxiolytic piece is a really, really big deal. So I'm studying that in myself and based on the literature I can find. But the reality is we've seen this increase of secondary metabolites across the board. The terpenoids and the cannabinoids on the new data, I have peaks everywhere. And especially as we bring in the field of photobiology and we work in an advanced way with organic agriculture, we can really, really boost terpenoid production. But the reality is when I look at the versions of inbred long-term cultivars that were in one region, I have many examples of those in our chemotype database. Essentially, I've seen a pretty large THC peak. I see a little bit of myrcene and almost no terpenes, almost no other cannabinoids. I find this very interesting. I think that's what's happened in this incredible incredible outbreeding project on the West Coast, is we've compressed the evolution of the plant in such a way that it has resulted in an incredible preponderance of secondary metabolites. And that's one of the exciting things in this moment, is to actually be studying what's showing up in the plant and what factors of production are resulting in that. What variables in the epigenetic, like epi as in outside of genetic influences. And looking to those is really my passion in this last period of time. So yesterday I was on the phone for 2 hours with one of the leading photobiologists in the world. He's an incredible researcher who introduced in short order some greater depth to inquiries I've been working on for a couple of years. And we're starting to find things out about the new technologies moving forward. So we've done a little bit about, just to quickly go back for 1 second, I want to do away with this whole sort of, I've got that strain, here's this phenotype, like actually genotypes, phenotypes, chemotypes, but then this new taxonomy, we've got narrow leaf and broad leaf, both drug and hemp cultivars and the ancestors. Then we've talked a little bit about sort of some of the evolution of the plant, but now it's this piece about some of the technology that we have in our midst. How am I doing on time, by the way? Do I have 10 minutes? Wonderful.
I think we're doing fine.
Cool.
Yeah.
Okay, so the photobiology study is a field of research that I believe will be a very fruitful inquiry for all producers to lean deeper into. And there's an incredible set of tools that have just shown up in the industry that are going to be available, that I've been working to make available. They're not the hobby crap that we find in sort of the LEDs that are being offered into the grow industry. The fact is LED lighting has changed the world. Last year, a Nobel Prize was awarded to a Japanese scientist for the invention of a blue LED that essentially emits the most true white light we've ever seen. It surpasses any modality of light in terms of the both bandwidth of spectrum and frequency, as well in the directional ability of these diodes. So a couple of things about lights, as LEDs are really changing everything, and I put a lot of faith that a big part of our future going forward will involve supplemental lighting in greenhouses as well as vertical farming. It's going to happen because climate change is happening really, really fast now. We know that California is in a world of hurt, and that 80% of the water is going to agriculture, and that it's being squandered largely on livestock. And the reality is we're in a position where we have no more time to wait on really innovative solutions and practices that are going to preserve water and sort of be really the best stewards that we can with the limited resources we have. We have to get pretty radical. So one of the things I'm aware of is in Austin, they have a 17-story tower that's got pink light facility where LEDs are lining these metal shelves, and there's robots that move giant shelves of plants out, and it looks a bit like the vertical farming that's happening in Japan. This is going to be a modality for food production, no question, because when we have 10 years of drought and we're waiting for that La Nina, El Nino to switch, and then rain and floods, suddenly we're going to have crop failures as the norm. When we have no water in our sort of water table to draw upon, we're really in this moment where we have to get really innovative. I believe there's a connection between food and medicine production, and that cannabis is about to make its world debut as very effective, safe, modern medicine. That those two things, and that cannabis can be an engine for driving forward a new kind of innovative approach in our agriculture. One aside I want to add right now, as I noticed John back there, is that there is a certification process that I would really urge everybody to consider who's actively involved in cannabis, who is organic and seeks to follow the organic path of production, called Clean Green. And this is the third-party certification that I believe has the best valor and possibility of upholding the integrity needed at this juncture to do what an OMRI certifier would do, to actually evaluate from the farmer's perspective, to come into soil samples and interview the farmers and build relationships and essentially go the route that OMRI has worked for years. But we cannot use organic because that's a term that's owned by the government. Organic, was actually swallowed by the USDA sometime around 2001. I was really involved in the organic standards movement as that was happening. But it is the case that it has been swallowed, and that you'll get an $11,000 fine minimum if you actually sort of market your product as organic. But here we have a key distinction that must happen in the industry. This is not going to be a popular perspective, but I believe this to be the truth, and I'm really willing to look at the truth, as uncomfortable as it is. And it's that we have on one side hydro and mineral salt fertilizer production. On the other side, we've got organic crop production. They couldn't be more different in the long-term implications of, especially as we're moving in scale. As this industry moves in scale, we've rarely ever seen a crop that's more heavily reliant on NPK in a mineral salt fertilizer perspective. And we're creating mountains of waste that is actually killing the Earth's waterways. What's really true is the fertilizer industry is just a disgusting mess and has brought more peril and havoc upon our ability to create a livable future than all but a few other industries. And that I really believe that there is an absolute necessity of coming forward and recognizing the implication of 55% of the world's fresh waterways being populated by algal blooms as a result of nitrate and phosphate contamination, that we cannot, as we scale, move into large-scale versions of that kind of economy and expect that to last for any amount of time. And meanwhile, we're actually going to demonstrate what I believe, what I hope, will be a little bit of a line in the sand around patient health, and I'll talk about that in a moment. Then on the organic side, and actually, do you want to?
Yeah, I got a question.
Yes.
Okay, back to the light. So we know that with supplemental lighting, we can take an outdoor plant and make it look indoors. What is the wavelength that's doing that?
So I want to talk about photobiology in a little bit greater depth and talk about the tools that are going to help us define that, because I believe a lot of what can be known right now about the wavelengths that are useful, and we're looking at both wavelengths for primary metabolite production, like photosynthesis, and we're looking at wavelengths for secondary metabolite production. But what's really true is we're stepping into an unknown. there's a way to work with the study of photobiology that is going to be a very evocative force as we're seeking to produce more effective and safe medicine. So I'll get to photobiology in greater depth in a moment. I know that's some of the technical bit. It's just that I wanted to celebrate that this difference between organic and mineral salt fertilizer production, I think is it's not clear to growers who are just simply reliant on a mode of production that's been convenient, that's produced a highly potent product, and it's very effective. The reality is I've, for a long time, done all of the versions. I have in a garden right now a mineral salt fertilizer test case. I have 4 different organic experiments going on, and I'm working on sort of data on all of these. But the truth is that mineral salts are very good at what they do. It's just that the cost we must pay in order to use those forces is absolutely tremendous and unacceptable, especially as we're growing. So I really believe, especially in Oregon, if you're not aligning your production at this point to be sort of in alignment with organic values, there's going to be a really detrimental hit to your business. And there is going to be a raising of awareness about the distinction between the two. Part of that is because I'm working to do something that I hope will have an impact in that. I have a company, I'm involved with FARMA Dispensary, but I also have a company called Nucleus Nurseries. And in this company, I have a PhD plant scientist who's now directing our research and development. We also have a biologist and a botanist who are working towards tissue culture. And that is, I'll talk about tissue culturing after photobiology. But one of the things that we've realized is we've got these great resources to generate some useful data right now. Everybody that I've talked to, including owners of some of the largest organic fertilizer companies in the world, have always incited this thought that if you have a mineral salt fertilizer-produced flower, you're going to have a flower that will taste harsh, it will taste nasty, it will be lower terpenoid. And I want to tell you that's not all true. It's just not. I've been measuring the flowers. And actually, in my long-standing organic heritage and all the different super soils I was doing, one of the giant shocks happened 3 years ago when I finally got lab results. And suddenly, I saw that the potency on my organic had very low terpenoids. It was wonderful. I loved everything about the process. But this mineral salt fertilizer was something like 2 times as much terpenoid and cannabinoid content. All of a sudden, I'm boosting strains from 22% to nearly 30%. This is true, so we can't be naive to the fact that mineral salt fertilizers are effective in engendering secondary metabolite production in a very robust fashion. But we're going to create a study now because I believe there's a patient health issue. It's a really prominent, serious concern, which is that we know cannabis has been used in the past and well studied for its ability to do phytoremediation. You can plant cannabis in a field, a brown field where heavy metals have accumulated, and use it to drain those metals out. There is a study that we have that indicates that copper in a soil environment was brought into the foliage and into the flower, and it actually was in the trichome itself. So cannabis is a very effective collector of heavy metals. And I believe that it's likely that the mineral salt fertilizer produced cannabis will likely have a higher level of heavy metals. And this is the thesis that we're starting with. And that the organically produced flowers will likely have a much lower level, even though there's a lot of heavy metals in organic production. It's more about how they're uptaken and the whole modality. So we're going to start this research, and we need to do 50 repetitions to actually be able to say something that will have weight as we go forward. But I believe if we can demonstrate this, this is going to help the patients to start to recognize that there is a distinction between something that has been grown on this incredible liquid mineral fertilizer program where the phosphate is based on urea formaldehyde, and when they're making the rock phosphorus into a water-soluble form, they're using huge amounts of sulfuric acid. And this is creating all kinds of problems outside of the phosphate and nitrates leaching everywhere. And that's true for all of it. There are no exceptions in mineral salt fertilizers for those issues. Also, if you want, sincerely, look at the Wikipedia page for fertilizers and just read the history of fertilizers there. There's a very succinct narrative that's well substantiated that describes this in greater depth. But know that's there. But then, over here in organic production, I believe we're going to actually need to use some new technology and sort of support to, in a way, create flowers that actually are more desirable in every way, have better secondary metabolites, have long-term ecological health as an implication of the way we're producing it, and that clean green is going to be a great resource, both certifying that you're doing what you say you're doing, but sure also providing some kind of relationship and education about how to do that. So I really want to say that bit of research we're just now initiating. It will take a long time to offer, but we're at work, and that's one of the things Nucleus Nurseries is involved with. I hope that we'll make some distinctions. Yes.
Is there an industry term outside of organic that we're using to show this?
I really believe right now clean green is the very best version, and that they have actually momentum. They've been well established in California, and it started by a person who was an organic certifier for a long time, and literally takes the same program and basically offers a clean green version. There are going to be other third-party certifications coming forward, but the truth is I see momentum there and integrity that I want to get behind, and I believe will help the industry. I haven't seen anything else as well organized or presented.
So you would go like into a dispensary, it would be like clean green, OG Kush.
One example, if you go to Oregon's Finest. Oh yes. So she's asking, is there another certification other than organic, or that we could use to call it organic? And I'm responding that clean green is that. And that if you go to Oregon's Finest, for example, you'll see many flowers produced from So Fresh farms that have clean green certification. There are many other producers coming forward. Here's the truth. It costs something. It's extra cost. And then it's this issue of we don't have any price incentives in the market at all, and there's no margins. We have the lowest priced cannabis anywhere in the country, and we're also producing a lot more than we probably need. There's this issue about this is an ideal we must strive towards, and I understand that the financial reality for most of us is such that it may not even be practical to get certified, but I just want to present this as part of the future that I see as being absolutely key in defining the quality in Oregon cannabis. Okay, so
I was just saying, yeah, I mean, clean green's not the only certified kind. It's another small operation out of Eugene that I've investigated. It's thumbs up, but it does cost money, but it's in the long run better than calling yourself organic, even if you are organic.
That's the key issue.
Yeah.
What I really want to say, he's saying there are other third parties, including one called
Certified kind.
Certified kind. This is, any of these are better than calling yourself organic, and that is case in point. I go to Denver or in other places, and there's organic everywhere in giant like plexiglass letters. And I go in, and I'm literally looking at their garden, and they've got mineral salt fertilizers everywhere, and they've got giant organic block letters, and everybody's organic. And it's sort of this diffuse, lackluster term that also should be being fined, and will be fined at some point. That I believe the way that we defer is go to other organizations. It's just that I have not seen the momentum, and maybe it will be the case that it will all change anywhere that I've seen with Clean Green, and I believe that will be a really wonderful infrastructure going forward. So that's just my personal perspective. Okay, that's a little bit about one of the new kinds of testing also that we're going to have to look forward to, which is actually becoming conscious about the heavy metals presence. Because, well, I'm going to leave testing issues to the master. So I would like now to do the photobiology piece. And so we'll do these two pieces here. Photobiology really is, even to know that word and know where to begin the search, is a key term as we're going forward. So I approached a company called BML Lighting from Austin, Texas. And last year, at the beginning of last year, found this company called Illumitex. Illumitex had an innovative new diode called the Serexi diode. It was the most efficient diode that's ever been released. And what's true about the sort of semiconductor industry driving innovation in LEDs is that for every year for the last decade, we've had twice the output for half the energy. That's continuing. Anybody who's jaded or sort of thinks that LED results are lackluster, that means missing that there's this incredible dynamic ride and we're not at the apex of that evolution. Right now, Illumitex has fixtures that are profoundly different than anything else I've seen in a conventional grow environment that's marketed for our industry. And if you read their website, you find out they're actually made for controlled environment production. But they also have formulas of lighting. They're not even going to suggest that you should know what frequencies you would want to work with. They're just giving you this sort of down. Here's the F3 for vegetative growth. Here's the F6. This is all wonderful, but this was their concession to a horticultural mode of production for these kind of LEDs. The founder of the horticultural aspect of the lighting in Illumitex left and started his own company, BML. And I'm tracking them because there isn't any technology I found that's rivaling them. They have research-grade LED fixtures. And one of the amazing things that we can start to do, now that we have these tools for the very first time ever, we can surgically isolate unique frequencies of light. So just to speak about light for a moment. Light exists in terms of lumens, in a way when we're measuring between 400 and 700 nanometers. What we see, what humans see, lumens are for humans. The PAR is for plants. PAR is the photosynthetic active radiation that the plants need for photosynthesis and the primary metabolite production, and also we see effects with the red and deep red that involve influence around secondary metabolite production. But the amazing thing is that now we've got the ability to actually split out all kinds of different frequencies. So if you go to BML's website, just as an educational tour, on the website is a tremendous amount of data, including peer-reviewed papers, and the tool that's really fun to play with, that I've been playing with a lot lately. It includes this ability to create your own bars, to mix your own spectrums, and to begin to research them. Okay, a couple of ways that light is being disseminated, though, that I want to distinguish LEDs between other sources of lighting, have to do with the fact that if you take an incandescent bulb, a fluorescent bulb, a high-pressure sodium, a metal halide, any kind of HID lighting, a plasma, induction light. If you look at all of these technologies, these are broadcasting photons in a 360-degree environment, right? It's shining from every direction. We're doing our best to corral those photons and get it to where we need. But in doing so, we're losing a huge amount of light. It takes energy to make the light, so one of the big efficiency losses is actually generating this field of light that's not where we want it. You can create hoods and have all kinds of reflection, you're still losing a lot of light. It's not efficient in that sense. The distinction in the light between output light and LED is that you now have a diode that's focusing the light in one direction only. So this is a huge efficiency improvement. I mean, you're not going to create free energy. We're still using energy to make these photons. But with the semiconductor approach versus burning gases, ultimately in our tubes, what we've got is this ability to focus an exact frequency of light in an exact range at a certain intensity. It's highly dimmable. And then we can get into this incredible nuanced version of lighting. So I want to excite you about the possibilities of playing with lighting more than, if you're involved with producing cannabis, more than you imagined before. Not just buying fixtures and hanging them over your crop, but everybody who's growing cannabis should be experimenting with the different kinds of light. So one of the ways people have been working with it recently is that I now am aware of many different epic scale producers who are using 10,000 Kelvin bulbs for the first 2 weeks of bloom in a mixture with the HPS bulbs, and then 10,000 Kelvin bulbs again at the last 2 weeks. And what's clear is that's really helpful. The 10,000 Kelvin is far on the low end of the blue spectrum, including some UV light. And that UV light, going back to the evolution of the plant, clearly is an aspect of how we increase rate of resin production. So there's no question that experimenting with UVB light, which unfortunately is more effectively emitted from fluorescent fixtures than from light-emitting diodes, is one really useful thing. But UVA light, which the LEDs emit really well, does a wonderful thing for plants. I've been using the Lunatex bars in a series, especially in one grow hive, in a series of different experiments. And the kind of development I watch happen under that grow bar, it's a single strip with about 8 diodes on it. It's 48 inches long and it's hanging 5 feet above the canopy, which is a huge distance for light to travel. In the HPS world, that would diminish so significantly that there would be very little light at the end. The LED is directing it so intensely that the plants are growing incredibly lush, and the internode length is much more stacked. We know that in the world of the influence of photobiology, there are phytochromes and there are cryptochromes. Cryptochromes, crypta as in like hidden, are relating to light that is received in the blue end of the spectrum. The phytochromes are more in the red end of the spectrum, between 680 and 730, more like 400 and 450. And these both have different functions in the plant development. So if you're focusing the blue light on the cryptochromes, you get leaf broadening, you get stout, compact internodes, you get lush vegetative development. But if you start to send the signals to more of the phytochromes, then you get stem elongation in long day plants, or no, excuse me, short day plants, which means they flower when you have a long night, then you also get flowering. This is how the photo period is indicated. One of the key insights in photobiology is that there's an interaction with these two phytochromes between a PFR and a PR, the two aspects of the phytochromes. PR associated with the near red, the PFR associated with the far red. What happens is when the plant is receiving the far red, as I was describing earlier, when the plant is shaded and then the forest floor, and now is getting signal to elongate, to grow lanky and reach for light until it outcompetes what's around it, that's also going to have other effects in the production of photosynthesis. There's a way in which the plants will take the PFR and convert it to PR. And this will go back and forth and equilibrate in the plant, and this is one of the engines of photosynthesis. So red light is really the most important light to talk about. It's maybe as simple as this. It's that if you look at frequencies of light, when you're far up on the frequency spectrum, like 700 nanometers, the light is moving fast with short wavelengths. And those short wavelengths are essentially just more active. So you get a way more efficient output when you're using red light than when you're using blue. It's literally just because of physics, a vastly more efficient source of a way to grow plants, to use less energy. So essentially, using the red light alone, supplementally, when you're growing outdoors, well, you have a blue sky with blue and green light dominating in a spectrometer if you're measuring sort of ambient lighting. And giving a nudge, in one study in the Netherlands resulted in a 75% production of boost secondary metabolites, specifically in strawberries, 75% extra vitamin C. And what they did was shine the red diodes exactly at the nodes of the plant, and the theory is that it's concentrating auxins and that the plant's pre-flowering, right as the nodes are developing towards setting blooms, is having the influence of this red light, is having a tremendous impact on the production of these metabolites. So I do believe that intercrop lighting, separate than above crop lighting and overhead lighting and indoor lighting, is a new consideration for everybody in every field. Actually bringing lights down into the canopy and these new bars that can actually diffuse effectively and bring less light intensity, more light frequency. It's actually a simple nudge. And when you're getting that red light, according to the most active and efficient light, it's having a great result. So that's one of the places I'm wanting to start working.
What red range are you talking about?
Specifically, you don't want far red, you want near red, so 680. That's one of the frequencies I recommend right away. 680 is near red, 730 far red, and 680 is going to be the primary juice for the photosynthesis. And that if you're essentially doing that, okay, so here's another experiment that happened. Last year I was down visiting a dispensary in San Francisco called Spark. They had initiated a bunch of photobiology experiments, and it turns out they'd done this with BML's fixtures. Because there's no one else making real technology that actually is meant for the industry at this juncture. That really is the case. I've reviewed Kind and Lush and a variety of other LED lights, and they all have varying trade-offs. This is really professional kit that will last for 10 years and never diminish in output. And is meant to be sprayed down with a hose and has no fans. And it's really amazing technology. They had this project going on in their basement where their goal, they had a PhD plant scientist who was there on staff for about 6 months. Their goal was to figure out if you could extend the length of bloom by using the red light at the end of the photo period. So instead of having 12 hours of light and 12 hours of darkness, and it is the darkness that indicates the photo period. So if you interrupt the darkness with a flash of red light, you will stop flower response. You could have 12 hours of light on the plant, just shine the light for just 1 second. That will stop flowering. That's all it takes. Because the PFR-PR conversion is going on, plant gets the signal, not time to flower, starts producing in that direction. That's my sense of how it goes. So what they're talking about is instead of shutting it down at 12 hours of light and bloom, go all the way to 14. But the last 10 minutes of 14, use deep red, or near red. 10 minutes of intense near red, and their idea was that this will reset the phytochromes. This will actually set the plant into rapidly being into the state it would have been in slowly after the lights came off. But now you've got more time to produce flowers and more photons to do that. But you're not confusing the photo period indicators.
And not changing the photo period, so you're not going 14 and then 12, you're actually going 14 and then 8.
Exactly. So that's one of the experiments that just happened. Sadly, they didn't do a rigorous study, and their data is really not going to be useful. But it's a demonstration of the kind of inquiry that I believe photobiology makes possible. What we know is there's some real potential there, and they were getting results, but unfortunately it's not a strong enough result that we could ever report as sort of a meaningful, definitive kind of result. So that's one example of using a frequency. Another, UV light. Now we can isolate UVA and UVB frequencies, and one thing that's clear is you don't need it all the time. But I would like for everybody to at least have some relationship with the possibility that enhanced trichome production will occur when you're flashing UV light throughout the flowering period. This is one of the bits of the research that this particular biologist had been working on, and he said that in Colorado he observed a profound increase in resin as opposed to the plants that were grown without that flashing of UV light. So this is not a steady UV light. This is literally on for 5 minutes every 4 hours during the light cycle and during bloom. So another example, but I think what I'm really interested in is combining a number of different inquiries at once. And I've been doing a lot of this already with the Illumitex pictures we got. The problem was the Illumitex only gave us pre-digested formulas, which include mixed spectrum. I've not learned anything about individual spectrums because we never had the technology as an industry until now through BML. One thing I should mention is that there is a way to actually purchase BML fixtures affordably, and that we're working hard to negotiate this. These are people who are not selling to our industry. They're starting to recognize that there's a huge market, and they've had some forays into the market. But the reality is there are horticultural LED lighting masters, and I've not found anything that was at that quality product. I believe that Roots Garden Supply in Northeast would be the best, negotiating directly with Neil at Roots. He's going to be a bridge to BML until there's other outlets possible. So what you can do is order these bars that you're creating your own formulas, and I think having individual frequencies in many different bars and experimenting with them and sort of 4x4 tenths is one of the processes that nucleus nurseries is now engaging in addition to the tissue culturing, in addition to the research. So those are a few examples of specific frequencies. There are more applications. But I just wanted to say that photobiology as a field and LEDs specifically have a giant role to play as we go forward in cannabis production. I see greenhouses that are organic, that have supplemental LED lighting, that have independent root zone heating, that are run in a very efficient ecological fashion, producing the best cannabis in the world from the new genetics that we're bringing from the vast genotypic library that we have. This is where we're different than anybody anywhere. It's a really big deal. But I want to spend just a few minutes on tissue culture as an idea and technology and approach to genetics before I pass the torch to the gentleman on my right. So tissue culturing is a modality in horticulture that's been used to create exact duplicate of genotypes that then can actually be a sterile mode of getting genetics out. One of the big deals that happened in the last many years is pests and pathogens have been really brought upon our community on a level that we've never seen before. Masterful producers who really felt that they could best any pest or predator ultimately are actually having a huge time with broad mites, russet mites, and the variety of viruses that are at this point undiagnosable. The PhD person who's actually authored these viral assays for the industry has created a starting point. We're now going to begin to test for viruses. But just as a corollary, if you're producing hops in the Willamette Valley, there's at least 25 viruses that they're looking to. And the rule is this: start with virus-free material, and then don't grow where viruses have been present. I hear that counsel constantly. It's very frustrating counsel, honestly. It's not what you want to hear. But we can actually take this as a call to action as we're now growing into a mature industry and recognize that we can actually, through tissue culturing, uniquely achieve virus-free cannabis. I think this is going to be a big deal because I believe they're playing a bigger role than has been reported, and they follow seed lines. So the viruses, just because you start seeds in a nucleate facility doesn't leave you free of the problems that can happen when a virus shows up. The tissue culturing, as in orchids being brought in, bromelia brought in from the jungle, they tissue culture these things 7 times. And here's how it works. You take an apical meristem, which is the newest growing shoot of the plant. You sever that, you bleach that, you alcohol that, you put that into a petri dish where agar and a variety of vitamins and minerals exist, and you cultivate that. And you essentially do this about 7 more times, then the apical meristem of that. And each time, what we're doing, we're in a race with viral RNA. We're trying to beat the path of the viral RNA, which is seeking to occupy the DNA of the plant. And that newest growing tip is how we're hoping to avoid that. But this is a means of also avoiding all other pathogens. So having not only the ability to potentially outrun viruses, which is still untested and we're working on passionately, but then also to have pathogen-free plants in general. This is a very exciting technology. However, what's really true, back to we're cornering ourselves genetically, is that if we create an industry that's built on tissue cultures and on clones, if we're mass disseminating shared genetics, as has been the case ever since Harborside opened in Oakland, that has been the great clearinghouse of genetics on the West Coast for many years. They're doing, just to put it into perspective, about a third of their business in clone sales for the last many years. And the reality is that this is actually an incredible sort of place where plants exchange every imaginable pathogen and pest and then go everywhere. And so we broadcast those things everywhere, and it's actually been a really sticky time for a lot of people who were used to gardening with occasional neem sprays and having that control whatever problems happen. So the issue with tissue culturing is that if we're relying on clones and tissue cultures, essentially we're halting the evolution of the plant. It's not everybody's job to continue to breed the plants and to continue to follow the path and progression that it is on. But it is many people's job, and I want to urge people to consider growing from seed a lot more. Because here's the reality. Even though you've got unknown genotypes, you're now creating males and females, hopefully, because you don't want feminized seeds, especially in the breeding program. You absolutely want these XX and XY combinations to continue to adapt, to evolve, and to create and proliferate incredible genetic diversity going forward, because this is our inheritance. We're starting with the greatest richness of known genotypes anywhere on the planet, and we had better end with even more and better, so that when climate change, when ice ages happen, when unforeseeable events over many generations occur, cannabis is still here for future generations. But it hasn't all been cloned into that one sexy Girl Scout cookie cut that everybody wants, that is the only thing that will sell because it has a certain THC number and is all genetically consistent. It's that now we want to introduce the kind of range and palette that Oregonians are real familiar with. Unbelievable diversity and nuance and craft, with producers that have not only their own programs for epigenetics and fertilizers and how they're processing the plant, but also their own genetics, because they're following the path into novel places. We will see very exciting things. That said, I want to say one last bit about genetics that I think is very important, because I don't know if people represent some perspective here. I want to say that there's an offering that's being made into the industry by a company that I feel passionately about, that Phylos Bioscience has been able to, beginning to author the very first phylogenetic tree of cannabis. We're about to be debuting into a kind of insight that no humans have had before. This is a very exciting time of discovery, to make connections, to look at what are the relationships between all of these hybrids and genetics. Where do they fit on the tree? How does that work? And it's not exactly a tree as much as an incredible way to visualize and organize this incredibly complex data. And that is happening in our backyard. And it's also filled with genotypes in our backyard. But further, there's a day coming where we're actually going to be facing serious issues with biopiracy. And this is a real concern. If you want an example, look to organic farming. If you have BT-resistant corn that is patented by Monsanto, and you're growing an organic farm across the street from the BT corn farm, we've already seen how this goes. What happens is Monsanto's lawyers sue the organic farmer to the ground, destroys their family farms. It's wiped out a whole lot of farms in the last decade, and actually won every case. They can out-lawyer any other farmer. And the reality is, what we need to protect ourselves from what is certainly to arrive, kind of patented, genetically modified iterations of cannabis that are coming, whether we like it or not, from one place or another. And that ultimately, we have at least the ability now to catalog and curate all the genotypes and all the special plants that exist in our region. And that if they were put into the public record, and the best way that I've ever heard to do this is to send a sample to Phylos Bioscience when they're accepting samples again, and then ultimately to put it into the public record will then protect, in a sense, that cultivar from being patented by another company that's going to come down the road shortly. This is actually a real concern, because we have special things here. You know, for example, the cultivar that's producing 30% THC and nearly 100 milligrams per gram of terpenes at just a shocking virile plant. It's doing roughly 2 to 3 times the amount of terpenoid combinations that anything else I'm growing, terpenophenolic compounds across the board. So that's the kind of thing, that's a freak show. It needs to be protected. It's special. It helps a lot of people. I don't want Bayer owning that, suing everybody else who's been working with it for a long time and laying claim to that. So I just want to celebrate that there are many facets of science all converging at this moment, and we're absolutely blessed to be in the position we are, where we get to interact with the absolute cutting edge of that. So that's it. I hope we have time for questions after Jeremy has had a case.
Give it up for this guy.
So, Jeremy's one of my favorite human beings on the face of the planet, I would admit. So I think what we'll do, I want to make a couple announcements. Let's take I don't sit still very well. We have beer, we have wine, we have champagne. Let's take like a 5-minute break. We'll come back. We'll listen to talk about testing and lab science. But before we do that, I do want to say a couple things. Because I know we'll lose you guys at the end. So, I guess I want to do a shout out to a couple people here. Tyson is in the back. He's been helping with this association from the beginning. He is from Oregon's Finest and So Fresh Farms. Megan, Matt and Megan from Pure Green. There are a number of PAC members here who you can talk to if you're interested in what we're working on. Shane is here. John is here. He can also talk to you about Clean Green Certified. I don't know who else is. Neil is here, tucked back over there with the hat on, who's looking like he's looking for himself, but he's actually sitting over there. And so we've been putting on these events, like I said, now this year, and the goal of the association, and we were going by the Oregon Growers Association, but we just feel like we need to be more expansive, is to offer these kind of low-cost events that are kind of in the weeds, talk about things that matter to you, talk about things that will help you move forward in this new industry, at least new in terms of recreational. And so I'm hoping that you consider joining. It's not very much. You can join on what is still OregonGrowersAssociation.com, ORGrowersAssociation.com. Charles is in the back. He is redoing our website. It will have legislative updates. It will have calendar dates. And these events are free for members. Like I said, this is the 3rd free event for members. And I hope that you all consider doing that. I think that there's a lot of opportunity here for us to do really great things. The next event is going to be April 17th. And what we've been talking about doing for that event is, and I'll send out the same kind of things, the Eventbrite invitation, the listserv. If your name is not on an email list, like you're one of 4 tickets that got purchased, in the back we have a notebook. You can put your email address down, we'll add you on. Or you can join the association on the website and you'll be automatically added. That event is going to be something I've been wanting to do for a long time, if any of you know me, which is I would like to talk about, we'll do 3 panels. That's the plan. The first panel is going to be accounting, bookkeeping, QuickBooks, sort of practical, not, again, lawyers telling you the little bit that we know about how to make this business work, but real practical. How do you set your QuickBooks up? How do you do your bookkeeping? We're going to have Mike from 7 Points, who is an amazing bookkeeper, come and talk about what their process has been. They told me they were $3 off in their accounting last year for their accountant, which is pretty amazing for a business that's mostly cash. So we're going to do that panel. Then we're going to do a breakout session, dispensaries, growing. Neil has talked about with Jamin down from Southern Oregon. Neil owns Roots, and which many of you probably know, and they're going to put on a kind of technical workshop for growers. We'll have a dispensary breakout panel, and we'll have a processor breakout panel, and you'll be able to pick which one you go to. And the final one will be a branding, marketing, PR breakout session with people who do this not for the cannabis industry, but for traditional industries who do things like central office. So I'm hoping that you all will want to come to it. We're trying to make it super cheap. If you're a member, it's free. So that would mean your membership would actually pay for itself by the time this next event happens. So we're going to try to make it cheap. We're not in the business of making money off of this. These are not for-profit conferences, but it's a way to bring the industry together. We are officially the oldest cannabis association somehow, maybe other than normal, I think, that's still working. So please help us and support us. Come to these events. We're super psyched to do them. And again, Tyson has been working on, Megan has been working on them. You can reach out to people. I know Chalice Farms is here somewhere. We collaborate with Mobley and Jeremy from FARMA and Jeremy from Cascadia. So go get a beer, have a drink, and then I'll call everyone back in like 7 minutes. We'll sit back down. Thank you. So I think what we'll do is we'll have Jeremy talk, and then we'll bring both Jeremys back up to answer questions if there are more questions. And then I know we have somebody from the new Women Grow group that is getting started or has gotten started who'd like to make an announcement. So we'll take from the floor. Actually, do you want to stand up now and do it? Sure.
Hi, my name is Sarah Batterby, and I'm a fairly recent import to Portland and also to this industry, but when I got here and I became involved in the industry, I was looking around for the ladies and how they get together, because that's what's happened in other parts of my career. And there's this amazing organization called Women Grow that did not have a chapter here in Portland, and so I decided to kick that off and round up a couple of people to help me with that. And we're launching it in, actually our first event was scheduled for May 7th, but I'm running into this terrible conflict with another event, so we're probably going to push it to June 7th, which is the first Thursday of the month. And you can find the event on Eventbrite if you just search for Women Grow, and if you go to the national organization, you can find my name and contact information there, also the Portland chapter. So, ladies, I look forward to seeing you, and men are also welcome.
So what Sarah's talking about, so Women Grow is a super cool group. They started in Colorado. They've got tons of traction, and it is a good thing. I mean, we have meetings all the time, and sometimes I look into like vast abyss of male faces, no offense. And I think that surely there are some women somewhere. So this is a great group, and it's really great to support women in this industry. And what Sarah was talking about was that we have started planning an event for Earl Blumenauer. When I say Earl Blumenauer, maybe she'll clap, because Earl Blumenauer is going to end he is going to federally legalize cannabis one day, God willing. And so we are throwing, the PAC and the association are planning, with the help of some super cool event planners, an event on May 7th. It will be cocktail attire, sit-down dinner. It's going to be amazing music, and it'll be at the Left Bank Annex. So we'll send out invitations, and we're hoping to show that the Oregon industry, like Colorado and Washington, that we can throw a better party than they can. And I don't know if you want to talk about Budtender's Ball. No, never mind. So we should all give Dope Magazine a round of applause. This is the Dope Show. Dope has an office over here. This is their space along with other people, and it's a great place for us to do this, and super affordable. So if you need an event space, talk to Dope. Jeremy, it's all yours.
Thank you.
And we're gonna still, if you want to stay afterwards, there's more beer, there's more wine and champagne, there's more pizza back there, and hopefully you guys can all network and talk to each other afterwards. So, thank you, Jeremy, and we'll have questions afterwards.
Thank you. Again, I just wanted to thank Amy, the Growers Pack, and Jeremy Plum for making my head spin again with so much good information. So I'm not quite sure how to follow that phenomenal presentation about kind of the genetics and background. I'm going to get into probably as what I consider as an exciting topic. It's going to be very practical and relevant to all of you folks participating in the industry now or have been over the last year. About lab standard practices, touch on that a little bit. What we may look forward to as far as kind of harmonizing those standardized practices and how we can easily do that over the next year to two that we're very likely going to see happen with the traction that the industry, the lab sector, as well as the general public has kind of put forward to Salem and other policymakers. So I guess one first step is to kind of understand what a standard is, how it works, both for a lab as well as for growers and for producers, because it's going to be across the industry. Not just the laboratories, but the growers, the extractors, the manufacturers of edibles. All of these individuals are going to have to follow some good practice or some standard of good practice. So what is a standard? We look at, from a regulatory standpoint, and I'll focus on laboratory operations. From a regulatory standpoint, there's two kind of areas where regulation can be put into our industry for lab operations. One bucket is the general standard operating procedures that are required for every laboratory to execute against. Good documentation practices, how you store and review your data, the qualifications of your staff. The second area of regulation, which should be contemplated completely separate, especially from a policy standpoint, is the list of tests that need to be performed and what those limits are for each test. So if you look at the first bucket, which is just quality standards and requirements that every laboratory in every industry has to execute against. And those standards are in place that we can easily translate into our industry. That is a must-have. That's a sunk cost of doing business as a laboratory operation. And it ensures public health and safety. It ensures that the data that you are paying for, that we are providing, is accurate, reliable, consistent, and that you can utilize that data to, one, protect you from any potential liability from bad data that you'd be using to bring your product to market, or two, make business decisions on how to adjust your process or whatever other decisions you may use that data for. That's just good practices. Where there's going to need to be extensive thought from a group of experts, analytical experts, plant toxicologists, medical professionals, is what the list of required tests for the different products are. We find ourselves with this set of rules that came out that is regulating the dispensary system. And I think we all, and anybody that's been participating in the industry over the last 2 years has seen the challenges that that's presented with having regulation just on the dispensaries. In my personal opinion, and what I would represent in the messages that I would give to the public and the policymakers, is that those retail dispensaries, they should not be responsible for the testing or the quality control of the product that they are putting to market. What they need to be responsible for is ensuring that the production and the manufacturing sectors of the industry are following those quality practices and are engaging with a quality laboratory. So where I'm kind of going with that separation of standard practices and standard tests that need to be required and the limits is those limits and the number of tests and how specific, if we have 200 pesticides or 50, that's where cost is being driven, is the list of requirements that need to be tested for each product. So if we're also able to move that testing upstream to the producers, the manufacturers, do somewhat larger batch testing with regulation on the producers and the manufacturers, which I think we all know is on its way, which is phenomenal, we're going to have a much more functional industry because we're moving that upstream and each section is regulated appropriately. So if we start looking at what those tests are required and how we kind of choose what attributes or what critical quality attributes for each product are, how is that decided? It's very hard. With our current set of regulations, they say pesticides, mold and mildew, and potency. Well, they don't say potency. They say pesticides and mold and mildew in the statute. The potency was added at the rule level. So that's really difficult first, because one, mold and mildew is the same thing, and it's completely ambiguous, so that was very difficult to deal with. Second, all of the different products should not be grouped into this one kind of hey, we have to do all these tests for all these products. It's cost intensive, it's not in the best interest of public health and safety. What we need to do, in my opinion, and that I would submit to the industry and any policymakers involved in this industry, would be that we need a group of both industry participants, regulators, and experts from different state agencies or different laboratories to really get together and say, okay, so these are the products that we have. Let's first define what the products are. Flower, extract product, which then you can break off, obviously, the CO2 residual or, to solvents, edibles and manufactured products. And then I really believe that Oregon is going to start coming out with these phenomenal new novel formulations of these products that we might not even be thinking about right now. And that's pretty exciting for me anyways. And more so, both formulations, and when I talk formulation, I'm not talking let's do an extraction and isolate THC or isolate this and make it pharmaceutical. What I'm talking about is utilizing those cannabinoids either in an extract or an edible or some other mechanism where they're delivering those cannabinoids in a more effective way. And we'll see where that goes. And I think the research licenses will help with that a lot. So looking at how we develop those tests, we want to look at all the different products that we have, define those, and then define tests for each of those. And that's where we want to consider that separately than a lab standard. So now we'll start talking about, I guess, what a lab standard is and how we would operate in a normal industry or how that would be regulated. The standard, there's a standard practice and there's a standard method. A standard practice would be something along the lines of every time I take in a sample, I have to document these specific items of the sample. I have to document it throughout my process. I have to store my documents for 2 years. Standard practice would be if somebody calls up and says, and this is where it gets kind of tricky because of the current state of the industry, there's even requirements in standard practice that if we have a complaint from a customer, whatever that complaint may be, that we have to start a report and evaluate if that complaint is valid, if there was a deviation in the standard operations of the lab, and the corrective actions that may come about because of that. These are all just standard practices that are defined in a general standard such as ISO 17025 or the TNI standard. And that's practice. A standard method, on the other hand, tells you specifically that you're going to test for THC in a flower with a GCMS. You're going to extract it this way, you're going to calibrate this way, your QC checks are that way. So that's a standard method. Those standard methods are not available really at all for cannabis or cannabis-based products. So that's challenging right there. But when we go back to the standard procedure, or 17025, or the standard operating practices, there is a standard procedure or practice for validating a new method in your facility. So that's what, any competent and qualified cannabis laboratory right now operating is, number one, they're developing the method in their laboratories. Two, they're doing significant work to show that they're meeting these attributes of a valid method to the best degree that they can within an individual laboratory. Those attributes of a valid method are repeatability, so that you shoot the sample at the same time, or a series of samples on the same day or different days, or a different analyst will prepare it, and you get a repeatable result ongoing. Accuracy is that if you're aiming at 10, you get 10, So the repeatability is multiple injections. The accuracy of a method is determined by analyzing a sample of known quantity. So typically this is done with a spike or some other mechanism in the lab. It could get quite technical if I start going down that road. But you're looking for a specific, you have a known number and your result is hitting that number as accuracy. Linearity is that if you have different concentrations that you're across a linear range, and because you're here, you're not going to have a different response if you're up here, so you're accurate across the range. Repeatability, linearity, accuracy, specificity is one where you're saying if I'm analyzing for THC, I know that in my chromatogram, so maybe I'll step back for a second. Chromatography, who's seen a chromatogram here? Okay, so I'll just get into basic laboratory equipment before I start diving into how to validate a method. So we're using chromatography primarily. Yeah, let's talk about how we analyze THC. That's probably a good start. So there's LC and there's GC, and I apologize if I'm stepping it too far back for some folks. But we have liquid chromatography and gas chromatography, and both of those are appropriate for specific aspects of the analysis, and that's where it takes a competent, trained chemist to understand which one they should be utilizing for each application. And we don't need to go into that argument. But primarily, any chromatography, what we're trying to do is take a sample that has a whole bunch of different analytes or compounds within it. And cannabis is very, very difficult as a sample goes because it has all these phenomenal compounds in it. It has waxes and all these different challenges from a sample standpoint. So we take this sample and we want to separate out all of those different components so that we can get a reading on what level each of those components are in that sample. We'll take that sample, put it into an extraction solvent, which essentially put that into a liquid, and then that is typically ground up, or if it's an oil, and then that solvent is pulling all of those different compounds out into that solution, into that liquid. We then take that liquid, do a series of filtration steps. And extraction efficiency is actually one of the most important parts of developing your method. For instance, an edible analysis or extracts with pesticides in extracts are very difficult to pull out and look at. Potent cannabinoids in edibles, because remember, we're trying to pull all those different compounds out of that sample into a liquid first. So the first step is actually drawing them out of that potentially tight matrix where they're all bound up. If it's a flower, you have to grind it very fine. If it's an edible, you have to get it very, very small and extract it. There's different solvents that you want to use for different compounds. So you develop that method, and then you have those compounds ideally with the best extraction efficiency possible in that liquid. You take that liquid and shoot that on your chromatography column, and that column will have these other chemicals in it that will react with your target analytes of interest and actually spit out a graphical representation as that sample is moving through your column with heat and temperature and pressure and all these other parameters that need to be developed and validated. Then you get a graph, essentially, and you separate all of the different compounds into what we call peaks. So you'll see a series of a flat line, and then it's a peak of THC, a peak of CBD, a peak of whatever we're looking for. And that's specificity, is that you can develop a method that actually is separating out all of those analytes of interest. So that's one other aspect of method validation. So that being said, I think that generally, there's so many different aspects of developing a method that it's not just, in this industry, we're not looking for, a specific pesticide in water that has an EPA-listed method that we can pull off the shelf, has everything that we need to do, and we can shoot it through and get it done. We're at the point, where some of this methodology is in the literature. There's some good cannabinoid potency in flower. There's some good terpene that is peer-reviewed literature that we based our methods on. The American Herbal Pharmacopoeia monograph, I'm not sure if anybody has seen that. It's a great resource, has some analytical methodology in there. But what we're currently doing now is trying to develop these methods in-house, perform our in-house validation, but until we start looking at this from a more holistic standpoint and say, we can't be one lab operating in our own sphere, in any lab, and say that this is a valid methodology, until we're able to take that same method and put that into somebody else's lab, and they can do the exact same thing and get the same result. And that's what interlaboratory validation is, and that's the next step in really robust analytical methods for cannabis. That's something that we're trying to initiate as best that we can in the current state of the industry. It's very challenging, as we all know. It's a competitive market out there, and there's a lot of proprietary techniques for analytical work, which I personally think there's nothing proprietary in analytical work. If you're an analytical facility, you should be completely transparent with the information and the methods that you're utilizing to generate that data, because that piece of data is the product that you are essentially selling to your client. That client should be able to see every step back until that they brought that in the door, gave you a sample, until they got that data out. That lab should be able to show you every single piece of that process with hard data and electronic data. And that's another part of a quality management system, is storage of data and documentation of data. And, you know, coming from the pharmaceutical backgrounds, you know, that's an environment where contract laboratory work is across the board, and contract manufacturing is utilized by all of the large and small industry participants. What I would love to see is more clients come in and ask me to see that whole sample, or say, and you don't even have to come in, just email and say, I want to see my data pack, because that data pack, you own that data pack. You don't just own that piece of paper with the number on it, you own how we generated that data. And that's the service that's being provided. So let's go somewhere else. Stop talking about that for a minute. So let's talk about, let's see. We have laboratory quality systems. So how do we implement laboratory quality systems to ensure that these labs are being inspected, that they are doing what they say they're doing? If we're going to put that we are operating to this standard, so we know what a standard is, and we're going to operate to that, and we have all these systems in place. Yes, we're doing that, but how is that recognized by a regulatory body or the clients or the industry that they're working in? And that's through accreditation. Accreditation is essentially an accreditation body such as the Oregon Environmental Laboratory Accreditation Program, the folks out of the Office of Public Health Laboratories, which I believe is going to be the state program that is going to be inspecting cannabis laboratories. It's likely that they will be looking at the competency of the laboratory to ensure that the laboratory is meeting these standards. Once that accreditation is completed, then the laboratory would get a license from the OLCC. This is my personal opinion. We still have to see what happens in Salem and whatnot and rules. But you'd be accredited to competency and in the ability to execute quality management systems, quality requirements, and technical requirements. And you take that accreditation and go to the OLCC and get a license, and then any licensed producer, processor, dispensary, extract, whatever the case may be, whatever those titles are, they would have an OLCC license, the lab would have an OLCC license, and there'd be a list. So every producer, processor could say, I want to know what labs I can work with that are providing this data to this level of quality, the standard that's required. So I believe that that's how it's going to happen. So back to the accreditation. We have our standard. We built the systems. We validated the methods to this standard, but we're just kind of in limbo. The next step is accreditation. And that's when we take all of those papers and procedures and policies that we've developed, wrap that up with a nice tight bow, and send it off to the accreditation body with a pretty significant check. They start reading through it to determine if against the standard, to determine that you do have the policies and procedures in place to meet the requirements of that standard. If they determine by a document review that you've met the standard in paper, they're going to come to the laboratory and start inspecting. And it's similar. I would say the inspection is very intense. I mean, working with FDA inspectors and European Union inspectors, it's intense. It's very stressful. They ask very hard questions. They're there to essentially find everything that you're doing wrong, point it out, and tell you to fix it, or you can't operate any longer in this industry. So it's a very big deal. And they're asking hard questions. They're going to ask you to see, let me see your logbook of customer complaints. Let me see your logbook of every time you failed the quality control check. Let me see your logbook of deviations. How did you fix these? What were the corrective actions? How many times did they repeat? Let me see your control charts. They're going to interview every single employee at that lab. What is your background? What experience do you have? How long have you been operating this method? Can I see your training file for this method? They're going to pick apart, then they'll go find, they'll say, let me see where we had a deviation. And then they'll go find that sample, and they're going to want to see, just as I said, that document trail from the time it came in to the time it went out the door, and every piece of information related to that. And that's the inspection process. Once you're able to demonstrate that you meet these requirements, you're awarded an accreditation. And that's not a one-time piece of paper that you receive. That's a yearly basis you have that inspection. On top of that inspection, at least twice a year, oftentimes more, you'll receive a proficiency test, unknown value. You have to receive that proficiency sample, and that same sample is spread throughout every other laboratory that's accredited to the same standard. So it's essentially a big round robin, what do they call it? Ring test. That's what I hear a lot of. Ring test. Essentially and it's not just Joe Schmo is going to send some samples through and see, how they turn out. It's very controlled. And that accreditation body is the proficiency samples are actually being sent by another organization that's third party that has another long list of standards that they have to meet. And I think that 17043 is the accreditation standard for ISO. So in any case, that's kind of the process of what a standard is, how we develop a quality manual and SOPs to execute against that standard, submit for accreditation, get accredited, now we can operate with a recognized level of quality. And I personally believe that is absolutely necessary for this industry, and it's very easily done with current quality management systems and accreditation programs that are available. Yes, sir.
Accreditation, so is this going to be done by a private third party, or is it going to be done by the state?
I think that's still to be determined. In my own personal opinion, I think that it's probably leaning towards a state agency or a state program, is my personal opinion. Yes.
Do you think it's time to get a whole state agency up to do that?
They've been doing it for about the last 6 months to a year, getting ready. Maybe 6 months. But they're actively engaged in developing these programs right now. And that's phenomenal because there's a couple folks within the state agencies that are spearheading this at the program level and are engaging cannabis industry participants to start learning how they need to modify their programs to best fit our industry's needs. And it's really phenomenal to see them being very proactive because they currently they are not charged. So this state program, they can't just say, oh, we want to start accrediting cannabis labs. They need statute, which is legislative law, and potentially rules to tell them how to do that. They don't have either of those first two steps yet, but they kind of recognize that they feel that it's going in that direction. So the legislative statute will say something along the lines of, the lab has to be accredited to this standard and test for these 4 things, and that's all it'll say. It won't have limits, it won't have who's accredited, it may have who's accredited, but the statute is very broad. And then there's going to be rules, and that's where we have an opportunity to look at, in statute is where I believe that it should be required for that first bucket of quality management systems is required. It's broader. The second level of rules is where in statute we may want to define what tests need to be done, but rules is where it's a much longer process, they're much easier to change over time. That's where we're going to get the group of experts together in committee and start defining what are those limits. What pesticide limits should be in play, what microbiological contaminants are of concern for what particular product types. Because we don't want just a blanket, you have to test this for everything. It's not cost effective, it's not scientifically justified. So once that happens, I believe that by the end of this year, there's going to be opportunities for cannabis labs to become accredited with I mean, we could get ISO accredited right now. The difficult thing is, with the current state of the market, no cannabis lab, to my knowledge, has revenue that's able to support that step to the next level of accreditation. It's very challenging. In any case, if you start looking at what those kind of test requirements are and what the limits should be, this gets into another really kind of, I don't want to say delicate, but an area of great contemplation, because I personally don't believe that flower should be required to be tested for potency as a number. I believe that it should be looked at from a requirement from a public health and safety standpoint, or if it's being directed for a direct medical benefit, then yes, it does have to be characterized. But looking at it from a THC strong or a CBD strong, or even breaking it down to this flower is 0 to 15%, this is 15 to 25, and 25 and above for a particular cannabinoid. The reason I would submit that position is because you have to look at the analytical variability, both the sample variability and the analytical variability of the products that we're working with. So everybody here would know that these plants oftentimes have great variability from this bud to this bud, and it's been well documented in lots of forums and lots of even peer-reviewed studies. Haskamp and Fischteek, I can't pronounce his name very well, a couple of folks from the Netherlands, they did a study, peer-reviewed, and looked at the difference of two specific varieties of cannabis, and showed that there's up to 25% difference from different points. And this is well documented with different points of data, different dispensaries, different growers, the same strain. And that's significant. 25, and I'm not talking 25 percentage points, I'm talking 25%. And that's where, if you start looking at ranges, where was I going with this? So the variety in the product itself needs to be considered separately not the variety, but the variation in the product itself needs to be considered separately than the variation seen in the analytical laboratory. Because the variation in the analytical laboratory can be controlled, the variation in the product cannot be. The way that we can attempt to mitigate the variation in the product is proper sampling. 3.5 grams from anything more than maybe a pound, 3.5 grams from anything more than maybe a pound is not relevant, in my opinion. And I believe that we can show good data that that's not. I believe about 1% of a total batch, and I'm not sure 1 to 10 pounds, anywhere in there. I think 1% of that batch is a reasonable, statistically relevant sample to look at to mitigate the variation in that flower product. So if we have good sampling practices, we mitigate that risk of variation. Then it comes into the lab. The first step is taking all those different flowers and getting them all into one pile and just grinding them up, essentially. Get them all into one big ground up pile, because say you're taking these different buds from or flowers from different parts, and they're all scattered, potentially 25% difference between them, and you're still getting a random sample because you want that difference. You want points of difference, and bring them all together, grind them up. And what we use a technique that's called a quartering technique. And essentially we take that 3.5 grams or 10 grams or 50 grams, whatever that sample is, grind it down, and this is flour. And it's not just, okay, now we have that sample ground up and we take our, small amount that we need to do that extraction I was describing. Like, there's another step of homogenization in the lab. Grind it up into a pile, quarter that into 4 pieces, pull 2 of them away, take those other 2 pieces, mix them up again, quarter them, 2 away, mix them, quarter them, 2 away, mix the remaining 2, until you get down to the actual quantity that is, we utilize 3 to 5 times the weight of what our analytical quantity necessary is. So it's not just take a bud and throw it in the jar. There's a series of steps to continue to reduce that variation or variability. In an analytical lab, you're looking at 20, in some environmental labs, up to 30%. You know, we try to have a 20% or less, you know, is reasonable in the scientific community. What we like to see in our validation for potency, we've looked at less than 10%, and those aren't percentage points. Those are, you know, if you're at 20%, it's 18 to 22 thereabouts. I just did my quick math right or wrong, I'm not sure, but you see what that's. It's not 20, not 10%, you know, not 10 to 30, but it's 20 plus or minus 1 percentage point or 2 percentage points. So that is also being driven, you know, that range is there. It's going to be there forever with this type of testing. And every step of the way, from when I weigh that sample to using a volume of liquid to extract it, to then using another volume for dilution, each one of those little steps has a slight little, what we would call measurement uncertainty. There's some level of uncertainty just because we're doing a test. I mean, it's inherent to the nature of the work that we're doing. So each of those little pieces start to keep adding up. And that's with the laboratory standard practices require that you measure and understand that uncertainty that kind of adds up throughout. And that's when we get to the end result, I can say this is 20% plus or minus 10%. In the upcoming environment that we're going to find ourselves in, your reports will be saying that. They will say 15% plus or minus this, or 10 milligrams plus or minus this, based on that validation project for that method, and more specifically for that product. So if you start looking at how results should be reported on a label or on a certificate, we really need to try to take the approach of giving meaningful information that is not too technically enhanced, I guess, to both the people operating in the industry that might not have the technical background, and also, more importantly, the consumers, the public, right? So what we're looking at, we're shifting into kind of this mode of really starting to modify some of our systems within the lab. And one of our first steps towards this accreditation and meeting the standard is looking at what are called significant figures. And that's with each of those pieces of variation that you have throughout each step, that's reducing the actual numbers that you could report on a report or a label. So what we're going to start seeing is it's not, you know, 20.01567321, it's 20%. And that's just as accurate, if not probably more accurate, than saying 20.01 or even 20.1 in the case of a cannabinoid potency. So that's where we're starting to shift in that direction internally. And as an industry, I kind of urge everybody to start looking at what do these numbers mean, and how are we interpreting these numbers? How can we give better information to the community, the consumers, and what do these numbers mean? Because just because there's more decimal places, I mean it's completely meaningless. And just because it's 16% to 19% on a flower, I mean to me that's completely meaningless. Even 15 to 20 is almost meaningless. Because I have absolutely no idea how that sample was taken, where those flowers came from, what the homogenization process was. But I mean, that's getting more into kind of how we can report that analytical information with extensive amount of work behind it. I guess that's the ultimate outcome that we'd like to see with laboratory regulations is in conjunction with good labeling practices and good labeling regulation, is that information can be used effectively by an adult that chooses to consume this cannabis product. If I'm going to go consume this product and I've never consumed cannabis before, I want to know that the data that I'm using to make my decision on this new, or if I've consumed it all the time, I want to know that this is clean and this is the dosage that I'm intending to take. And I think that's the most important piece for laboratory regulation to move forward with, you know, why we need it, you know. So, is there any topics that anybody wants to hear about? I was, yeah, there are questions. Do you want to come up and do some questions?
There's one piece that's so amazing. I don't know if everybody's aware of it. We're very lucky to have Jeremy and many other really passionate, talented chemists bringing all kinds of credibility to bear on the subject. Because, for example, when we're in the Bay Area, we see the preponderance of quanticam results. Like, this is a technology that Steele Halen is pushing, and while they can do great results in-house, they're actually suggesting that dispensaries should have these machines that do absolute bullshit results. There's a level, it's intolerable variance, and they're suggesting this is an okay baseline. And the fact is, the way they're describing is just so much further. This near-infrared technology and with some of the sort of consumer-grade analytics devices I've seen show up in facets.
No, that's a great example, this IR technology. I mean, every industry, pharma, dietary supplements, material science has tried to take a stab at using IR and other spectrometry techniques to quantitate different compounds. And none of them have been successful for the last 50 years to actually develop technology that does this very well. And these very well-established, multi-billion-dollar industries putting million-dollar labs together just to develop this technology, and they've been unsuccessful. Unfortunately, now there's this kind of opportunity for salespeople to insert this technology into an industry that is not informed of what they're actually looking at and potentially taking advantage of individuals.
It's a giant deal that the largest dispensary on the planet right now has data on the shelf that is all created with that technology.
I was actually talking about that at lunch, but there's another one that's coming out, and it's exactly that. The variability it's unacceptable from a good analytical standpoint. It might tell you if there's THC or CBD in there, but that's about it.
It's quackery.
Yeah. And that's all leading to variability.
I was just curious, are you guys developing any techniques to detect viruses?
We currently are not looking at virus.
We have. There's actually a viral assay that's been offered, but it's not been employed. It will be shortly, and it needs to become more broad spectrum. We're looking specifically at TMV-like viruses, but eventually it will be much like Humulus or hops viruses. There'll be 25 we're looking at, and it'll be important to look at that. So it's in the pipeline. We actually have assays offered. I'm not sure when that will actually be executed.
What type of assays?
Actually, this man who's the PhD director of research and development for this, he's offered one, but the truth is, I think it's going to be a while before it's fully implemented.
Talk a little louder, Jeremy.
It's going to be a while before the assays will be implemented and available to the industry, but it's very important to know working on it.
Is it chromatography based or ELISA?
Actually, I'm wondering, can you say anything about that?
Quantitative PCR.
Yeah.
It was 6 months.
6 months. We'll be looking to file this as well on that issue.
Jeremy, in your lab, you spoke of repeatability, and you mentioned that in your standard operating procedure, you kept the records for 2 years.
Right.
My ticket, I have been your client and probably will be again. Does it help to have repeat things? I make edibles, first off, so I'm even greater than the bud. Does that help build this database? And in part of the accreditation, and of course you can't speculate on what the rules committee is going to do, otherwise we would still really have safe access. Thank you, 3460.
Can you repeat the question?
Not you.
So I think it was essentially putting the same sample back through, or looking at a sample in one instance, and then if a manufacturing event occurs again, to do that the same process. Yeah, very, very important. My opinion would be that every single batch, be it a flower batch, an edible, at least within the next 2 years, I feel that every finished product should be tested, particularly the edibles, for potency. I do not agree with utilizing calculations from an oil and bringing that forward to an edible product. I know it's possible, but I do not believe that it should be happening at the current state, at the current juncture in our industry.
One thing I'd like to speak to is the variance in the flower samples. When I first found 3 years ago a sample that had 29% THC in an area that clearly had much better light intensity, and then a shadowed part of the plant, sort of 19% THC, just massive swing, it started to become clear that this is partly an issue of canopy management and of how we're addressing production. So back to LEDs, there is a measurement of light that we should all be well versed in, PPFD. This is a very important thing because what we're actually able to talk about now is how much the photons are actually directly resulting in photosynthetic activity and are in a usable, meaningful form of the plant. If you look at the newest technology on the BML website, you'll see data that shows these unbelievable PPFD numbers. So different than PPF numbers, which really is photosynthetic photon flux. PPFD adds a dimension, and it has to do with sort of, if I shine a bright light up here, it's casting that light everywhere, but it's not telling me how much I'm getting actually in terms of photosynthetic benefit to the canopy. But if we have much better lighting, that has better canopy penetration, and organize our canopies as such, then we can actually reduce the variance as producers significantly. But LEDs really are the best way to get even PPFD. And there's a lot of data about how you get to that, to measuring that well, but it's very important. We start looking at PPFD as the most meaningful measurement of photons.
And I want to just circle back to that relatively strong statement about not using oil to calculate in an edible product. I'm not going to retract that statement. I think it's what should be done. But also what should be done is looking at every single type of edible as its own method. And that is not very feasible in the current state of cannabis quality control laboratory operations. What we should be looking at is if there's an edible manufacturer that's moving forward with production and licensing within the new model of regulation, each of those products that that manufacturer is going to be making, and even from an oil standpoint or an extract standpoint, more specifically edibles, they need a specific method for each of the challenges that that formulation presents. Dairies, the different fats, taffies, caramels, they're all very different, and it's not a one-size-fits-all. We get back to that extraction step of when we first have the sample and we need to get those compounds out, or we need to grind that up. Certain things work better with certain matrices. So we're not just going to take the same thing we do for flour and it's going to work for a taffy. And just because it works for a taffy doesn't mean it's going to work for a cake, and it doesn't mean it's going to work for, it probably would work for oil. Extracts are pretty easy, but in any case, that's another challenge that we have, is because we need a method for each product and each unique formulation, and every manufacturer eventually will have, we currently do have with some of our clients that this is a method that we use for their product, and we use it for no other product just because we've developed that relationship, and that's where it's going to move forward with.
One case in point, a client in Germany has a product called Toro bars. The reality is that they're able to now work on getting to such a fine level as 2.5 milligrams in every individual cube in a multifaceted bar. This is a really great delivery for a lot of patients, because one of the first patients we found is that actually tiny, tiny minuscule doses can be the absolute life-changing quality of life explosion for many patients. There's others who just want to get high. It really, at Pharma, we're now meeting tons of people who 1 milligram or 5 milligrams twice a day is absolutely transforming their life. The psychoactive or the CNS effects are really in the background, and in the foreground is therapeutic relief and symptomatic control. It's amazing.
And don't get me wrong, there's significant costs associated with developing that method and developing that process, both for the laboratory and the manufacturer. But as we move forward with a more robust regulatory system on the manufacturers, there's going to be additional costs. And you put that effort in up front to have accurate and reliable results with your product, and you know that it's going to be consistent on the shelves. It's always going to have labels that are, you're going to have a better position in the marketplace as well, because you put a little bit of effort in up front. And potentially, if you start looking at it from a batch perspective, and you can demonstrate that it's the same time after time, then maybe you can start moving back to just testing the oil, doing a calculation out, and validating your process through analytical work, where it doesn't have to be just at the end, but you can validate a data set across your process and do what's called process validation to show that you can consistently produce the same thing time in and time out with a procedure that you have written in your manufacturing facility that you follow every time, and you log your date and initials, when you're making it, what products you're using, do your calculations. And that's how, essentially that's how pharma works. There are QC tests at the end, but there's a significant pharmaceutical industry. The pharmaceutical industry, and dietary supplements, and even food, they do a process validation to show that they can consistently achieve the same outcome with that process. It reduces the quality control testing at the end in a lot of cases.
So just on another tangent, as you talked about mold mildew earlier, do you have those particular ones in mind to go forward with, rather than just saying mold, numerous count? That's
Right.
not really appropriate. It's untied.
Yes, I think, there may, I personally believe that total yeast and mold count is not a good quality attribute for particularly flour. It may be counted, but I don't believe it should be a limit. There's no...
Especially if it's outdoors.
Yeah, and I mean, especially from looking at it from a yeast standpoint, yeast is, you're drinking more yeast in a beer than you would on a failing, potentially consuming with a flour that fails in the current state. I believe that we will stratify or segregate different, more pathogentic mold species. Again, the Cannabis Safety Institute, Phylos, and some of the folks have been working really well on gathering information on what those are. And also in food safety, we just need to look at edible products from what are the standards for that type of food if we were going to make it without cannabis.
Okay. One tiny bit about the mold and mildew aspect is that we found that actually aspergillus is the big nasty, and it's really a serious concern. It could be a very big deal for an immunocompromised person or for anybody. But one of the realities too is that it's very likely you'll get that in the bong more than you'll get that in the flour. In the bong, you have a watery environment which has actually impurities in the water. The metabolites attached to, so you're literally filtering out like THC or CBD, and they're binding to these particles in water. But then also you've got an anaerobic environment that's inculcating a fungal sort of anaerobic scene where nasty things can happen. And the aspergillus is the big deal.
Do you have a program for validating ball water now?
Dump it out and keep rushing.
I have a question on the organics that you were talking about before. Just to clarify then what you were saying, the cannabis industry is going to be, you see, regulated differently than food and beverage or dietary supplements by not solely going after the organics label and more towards clean green, you say?
We're in a position where until Schedule 1 changes, until the categorizing of cannabis being in Schedule 1 is recategorized, we can't use that term. That is a USDA term. And so the thought is, rather than wait years down the road where we can actually have a USDA-blessed terminology and not also then potentially be vulnerable to $11,000 penalties, that clean green is the best path I've seen.
Do you think that's years still? Years?
The thing is, there's a lot of speculation. My great hope in any more rational process is that this year that's going to pop, one way or another. The reality is, I have an attorney who's talking to me about, be ready for 10 years, who knows? We just can't, it's a giant, there's a lot of complexity in that process. And we need to know that now. Our patients need to know that now. I think there's really sensitive and important data and sort of like heavy metals or other concerns. We need to be able to distinguish. It's indistinguishable other than clean green or some of the other third-party.
Well, and I'm asking as a grower because I'm looking just 100% organic, and so I want to be certified, and so I'm not going to go search out the label. I'm going to search out the clean, at least for now anyway.
I would love for you to be able to contact Clean Green directly. There is a representative serving sort of from Medford North and then something in South Oregon. And the reality is, John, are you in the room still?
So, yes, there. So John is actually a clean inspector, and anybody who needs to ask about that certification process, he's here now.
John?
Yes.
Great, thank you. Thanks.
As a grower submitting samples for testing, what do you see the most responsible method right now? We do have a lot of variants throughout the canopy. How would you like it? What's the most responsible way to receive the samples?
Ideally, so this is idealistic vision. You would have a form to start with that says this is the date, this is the name, this is who's doing the sampling, this is where I'm doing the sampling, this is the number of containers that I have, this is what containers I'm actually taking samples from. We're talking about a batch sample. Get some documentation to your sampling event, and then procure, and this is well described actually in USP 561 is the Articles of Botanical Origin monograph from the US Pharmacopoeia. It actually, so these are articles of botanical origin that are utilized for a medical purpose, and it has a very specific sampling plan within that USP 561. So we look at, essentially you look at the number of containers, you determine how many containers you have to take it out of. If it's a significantly large number of containers, say you're doing half ounce, or you want to actually bring your product to market in half ounce containers or 1 ounce containers for whatever purposes. You take, typically, let's just look realistically. You have a pound. Take from the top, the middle, and the bottom. The smaller, probably the better. If we're going to request 3.5 grams for the sample, ideally they're about a half a gram each, because you don't want to throw in, a 2 or 3 gram product. You really want to take top, middle, bottom as random as possible. Essentially, it'd be best to have it in the bag already, kind of shake it around, get it in there, and have a glove on. If you have IPA, wipe down a little bit first, so you're not contaminating your environment potentially. Take those samples, get those into a bag. Most labs will provide you with a sterile sampling bag if you ask them, to say, hey, can I get a few bags so I can bring them into you and not, have to worry about it. We'd actually prefer that because then it's in a sterile bag. We know that there's no environmental contaminants. Again, just top, middle, bottom, as random as possible. If you want really a more detailed, extensive look at it for kind of regulatory purposes in the future, I'm happy to talk with you offline in more detail. Yes?
What are you predicting is going to be the standards for pesticide testing?
That's a very, very good question. So if I had a crystal ball, I would say that pesticides are going to be the most expensive tests that we're ever going to have to do for this product in this industry. With the current state of the industry and the current regulations, the pesticides are a bit absurd the way that they were defined, unfortunately. They're put into categories without key targets that we're looking for. Roger Volker down at OG Analytical has done a phenomenal amount of work, and the OG Analytical team altogether. He is a pesticide expert, U.S. Department of Agriculture, and looking at what is available at your grocery store, what's available at Home Depot, what are people actually using, talking to growers. I've been a grower for half of my life or more now. I kind of know what people use and what we should be looking for. But if more growers that are, producers, cultivators, they can help with that process, and Roger's done phenomenal work. What we're looking at is probably a list of about 50 pesticides that would be required for every batch. It's not DDT or these other compounds that nobody's been able to get for 30 years and are not very relevant to our industry, but it's, the plant growth regulators. It's some of the fungicides, these other common and modern pesticides that are typically used for ornamental plants and are very toxic. So we're looking at what's available and also what's very toxic, because if something's readily available, or it's not readily available and it's not that toxic, you don't really worry about it. So they've done, and actually the Department of Environmental Quality, the Department of Ag, they actually have lists. So Roger has taken, and you can see how many pesticides of a certain type have been sold this year in Oregon. So there's really good data there to help us define this. What's very tricky is that we don't have very significant toxicology information on these pesticides when they're inhaled in and the different effects that they have through that absorption mechanism in the lungs. We have to be quite conservative with that. I think that Oregon has set a significant precedent, at least with the 0.1 parts per million. To my knowledge, we were the very first state to actually set a limit for that. So that's a really big deal, actually, because essentially all of these pesticides that anybody uses, even an organic one on cannabis, is absolutely illegal, not because it's cannabis, but because that pesticide is not registered for that product. And that's a big difficulty that we have as we're trying to develop regulation. Essentially, these state agencies are looking at it from the standpoint to, if I'm going to use this pesticide on apples, it has to be approved for apples, and the manufacturer of that pesticide is responsible for saying, you can put this much on your apples and you're still safe. That has not been done for cannabis, so we're really trying to piecemeal all this information we have together and make a very informed and rational decision. And it's not going to blow up a $1,000 test, but we're looking at a very concise list of analytes that are potentially harmful, and then that's your required QC test. There may be an opportunity with the random sampling plans where, yes, this is very concise, but every 3 months or 6 months, you're going to get knocked on your door and an inspector says, I'm going to take your random samples now, you have to hand over what you have, and then you get tested for 300 of them. And that's really kind of those, there's either some issue going on or it's just a requirement for random sampling, and that might cost, $300 to $500 for, 300. The standards are quite expensive.
The regular agricultural ones, right?
Yeah. I mean, a typical agricultural commodity pesticide test is $300 to $500, but you're looking at a truckload full of product. So we understand the consideration of cost in this industry, and what we're dealing with the current state. So again, it gets back to defining what's appropriate and not putting some ambiguous, in any case.
So what do you test for?
There is one thing to say about pesticides, which is from a grower's perspective, there's no question the way forward is living controls. This is another case for why we should be more sophisticated about organic crop production. The reality is it doesn't work well with mineral salt fertilizer programs. That those are tantamount to using a chemical control process, because for example, the nematodes, which are going to control the fungal gnat larvae, will not flourish in the cocoa filled with mineral salt fertilizers. Those nematodes will absolutely annihilate all of the things that are nasty in the organic program. It's not even an option over here. I tell you, the way forward, we have to get in line with high quality organic production and all the living controls and the certifications that will help to give confidence to patients and consumers going forward that you're interacting with a farm or a producer that's working at this level, that cares that passionately, and that's one of the, when I look to brands in Oregon, any kind of other facet of craft industry, I think we see that same phenomenon, that I really do see this short future of being able to apply all these compounds that people have been using wholesale for the last decade, and it's really not acceptable.
So realizing these compounds are being applied right now, and it's probably going for some time, what is the current research being done and the timeline that we expect it to take to find out what impact plant growth regulators have smoking?
Yeah, I think that the impact that they have on the human body is well characterized once they're in the bloodstream. Yeah. What we're looking at is what amount actually gets transferred if it's on plant material through whatever combustion device or vaporization device, and there's multiple different ways to do that, obviously. What is the rate or the efficiency of that pesticide to get transferred through whatever device it is and into the lungs? Once they're into the lungs, it's great likelihood that they're going to be absorbed into the mucal membranes. They will be absorbed fairly readily, is my best guess for most pesticides. So that's really what we're looking at, is what the rate of transfer is for the devices and the amount that's being consumed. So once they're in the body, we know, what or I don't personally off the top of my head, but the scientific community has a significant amount of information about the toxicology of all the pesticides once they're in your body.
How it gets in your body, though, they impact that, whether that comes in through your immune system or through your lungs.
Once it's,
Whether you're feeding it and you have an actual
it does, because that will determine how much actually gets in your body. So if you take 2 of these and put this one in, the same amount, this one goes in your ear, you don't get any of it, so it doesn't have any bad effect. If you eat it, it's going to absorb through your gut, and you're going to have that bad effect. So it's really, you're exactly right. It is how it goes into the body, because that's the level that you will see in your body. It's the absorption of that pesticide through the different routes of delivery. The same way, edibles versus smoking. There's different routes of delivery and different absorption mechanisms.
One of the issues, though, with that is that actually, I think there's this sense of we're not going to see long-term health effects on something like even spinosad, or even like in the organic controls department, this phrase. At what point does azadirachtin become a toxic compound over the long term? It's just, I think cannabis is so unique in that we're bringing it into the bronchioles, we're bringing it right in the bloodstream. I'm not going to wait for that research because it's going to take a really long time, and they're disincentivized for producing it. Perhaps there will be all kinds of industry support to do that and have a clear insight at some point, but I think ruling out those compounds in general by having a better farm is a great way to go. I'm telling you this.
Next question is completely unrelated. I'm going to crack some seeds off, and they're supposed to be high CBDs. Do you guys do testing for vegetative state for CBD?
Yes.
What's the best way to bring those to?
Fan leaves, 3 to 5 fan leaves. We can give you a read if it's going to be THC or CBD. With great likelihood. It's not going to tell you this is going to be this percentage or this is going to be this ratio, but we can kind of see preliminary precursors of those acidic forms just in the leaf material, very low levels.
I did recently see a lab in the Bay sort of starting to make a claim that we're going to tell you what the chemotype will be from that early data and tell you that's not plausible.
There's a lab that's talking about it in Seattle, so they'll show you the spikes.
Yeah,
that's it.
Yeah.
So that's all I'm looking for.
And that's
eliminates 50% of the seeds right out the gate.
Yeah, that's a huge advantage instead of having to flower it out. As soon as you get a handful of fan leaves, you can take a look at that. Cool.
Are there any substances that, when added to oils, like you're making some malts, you have wax or something like that, that skew results in the process of the photography?
Yes, yeah, definitely. And that's where we look at specificity and extraction efficiency. So topical type oils, I think that's what you're referring to, those are very difficult, very, very difficult to analyze. But it's doable. I mean, it really is. When those types of products come in, it's really, okay, now we have to stop. We have to look at this one particular sample. It doesn't just kind of click through with the rest of them. And there's a much more extensive quality kind of control. And once we start seeing the same product come back through time and time again, then that's when we get to the point of having that, we're really now we're getting a method that has your own number on it. It says, this is HPLC analysis for XYZ product, we actually have that own method. But it's really looking at the extraction, what your extraction solvents are, what your column chromatography is, and ensuring that you have specificity. And that's done through method validation process.
So in that situation, it might be better to send you just the infused oil rather than adding the wax and that kind of stuff to it before it's tested.
Yes, but then you fall into that kind of testing before the finished product, so you don't actually, I could chat offline with you in a little bit more detail, but essentially what we're looking at is the more ingredients that you add, the more difficult it gets from an analytical standpoint. I believe that we can really, with the right equipment, the right know-how, and the right motivation, you can analyze anything accurately. It's just time and money to get there.
Do you have anybody bringing in extracts made with terpenes yet?
Extracts made with additional terpenes?
No, made with terpenes as a solvent, like industrial terpenes.
I don't actually see most of the splat samples that go through the lab any longer, but I can't say that we have seen actually using terpenes as an extract. I can't say that we haven't discussed it significantly though.
I will say in a production environment, terpenes are amazing pest control. There are many applications for just terpenes.
Yeah, terpenes actually could potentially be utilized as an extraction solvent. Yeah, definitely.
Solvents. And in fact, really a lot of the oils that are very, very high in terpene, like the holy water in the Bay Area, it just burns the back of my throat terribly. I mean, it's inhaling solvent. That's
I think the limonene was limonene.
What was that?
I think it's made with limonene.
Yeah, that would be what I would think.
Yeah, it's the mountains with that limonene. It's very profound. It's not desirable.
So do you use residual solvent testing, and where do you think that that's going to go with regards to concentrates, and really what should we be most concerned about? Because there's quite a few residual solvents that have hydrocarbon extractions.
Yeah, what are real safe levels? Real safe levels, right. Okay, so that's a great question, and a few parts to the question. Yes, in my crystal ball, residual solvents are going to be required for any solvent-based extraction in future regulation. So then, yes, we do test for them. The methodology is typically GC headspace, potentially GC headspace mass spec. And these methods are very well developed in other industries. Actually, when I was in pharmaceuticals, that was my primary analytical work, was residual solvents around solvent-based pharmaceutical processing. There's extensive literature and methodology and techniques on good residual solvent analysis. When we get into the cannabis industry, the analytical methodology is the same, but in pharmaceuticals, you're primarily looking for one or two solvents that you know that you're using in your process. You know what those potential impurities for that solvent might be, and you again have a valid method that's looking at a specific product, specific analytes. In this industry, we all know that there's varying grades of solvents that we can procure, and I would say as an extractors out there, buy the highest purity solvent you possibly can find. And I know that's very difficult because if folks don't want to sell, air gas and Praxair or Norco, they're very hesitant, and there's a significant background check to actually procure these solvents. I would hope that as the industry becomes more regulated, these markets become legitimized at a state level, that some of these local companies will more readily supply those medical gases. A phenomenal thing that we just came up with, that this is totally sidetracked, I'll get back to residual solvents. We recently, within the last month, identified a waste management company that will take all of our cannabis waste with a dossier out of our shop and log it for us. So, I mean, this is a nationwide company that operates in Washington and Oregon, and they're getting on board. They've realized the time is now. All these other businesses have these needs that they can serve, and it's not, So anyways, back to the solvent question. Safe levels. It depends on what solvent you're looking at. Benzene has to be less than 2 parts per million. Methanol, less than 5,000. And I'm using those references from a pharmaceutical standpoint for a very large, orally ingested dose. Pentane and pentane isomers are common with butane. There's a significant number of isomers of those heavier butane alcohols and some of these other kind of related isomers. The butane and propane are actually quite safe overall, and they actually come out very easily in most solvents. We actually see very low levels. But what is really concerning is in that chromatogram with those peaks is those late eluders, those higher boiling point solvents that are the impurities in the gas that you're using, or extractors are using. So there's these impurities, and they're a higher boiling point, they're heavier. It's sometimes like mystery oil I see, and it's funny because we can figure out what mystery oil is very easily, and we need to get that information out there to the community, to the regulators, that we can figure this out with the scientific approach and make it safe. But the trouble is with a lot of these unregulated and uncontrolled cleanup processes or purging processes, that it's a low temperature and it's driving off the propane and the butane easily, but these higher boiling point impurities are sticking around. And that's the same issue that we would have in the pharmaceutical industry. So it's really just looking at the process and and trying to control that with both purge and cleanup steps and good analytical work that has sufficient detection limits to show that they're not there. I would say that benzene is probably the worst impurity that we're going to see, and it has been documented in a lot of products. And that's carcinogenic, just gnarly stuff.
You know specifically about hexane or the molecule?
Hexane is 290 for a 10-gram orally ingested dose. Yeah, it's 290. It's pretty low. Hexane is bad.
We're good. Concentrated. Okay. So if you're testing a concentrated, you saw hexane.
It's bad news. Hexane is bad actor. Hexane and benzene are the couple of worst actors.
So it's 8 o'clock. We'll do one more question, and then there's a few more beers left, and so.
So butane is pretty much like the largest solvent by volume, and as far as what people use to make this, it's really easily accessible, and it's relatively pure. And that's going to be like the majority of what home users can produce right now. What level are you saying is acceptable as a residual? I don't really advocate for using hydrocarbons as a solvent.
Right.
I'd say like subcritical or supercritical CO2.
Right. I'll preface this statement with I am not a medical doctor or a toxicologist, and I cannot make a recommendation on a safe level for a solvent. But pentane, which is very close to butane, is listed at 5,000 parts per million in the pharmaceutical standard. What's that? For ingestion. But also consider, I would submit, briefly, because I know we're a time constraint, but I would submit that there's likely significant butane that is inhaled with a lighter to the same level as a 100 or 200 or 300 parts per million extract.
Thank you.
And that's been demonstrated in some pretty good research, actually.
Yeah. Thank you.
We really want to thank the Oregon Growers Association and all the integrity in our community, especially spearheaded by this amazing woman, Amy, who has actually really brought us all together.
Although there are lots of people in this room who participated in making this happen, I'm not going to call everybody out again. We are rebranding, so I don't have like a giant banner with all our information or pamphlets yet. But I do have, we're still the Oregon Growers Association online. It is not very updated. Charles is banging it out, and we're going to soon be the Oregon Cannabis Association because there are so many people doing so many rad things. If you have questions for me, this is really a grassroots group. I mean, we put this on because people ask for it. We put on the Tax Lunch and Learn because people ask for it. So if you have ideas or thoughts or things you want to see, I mean, we are trying to make this as much for this industry as possible. And I was struck when I was sitting here listening to you talk about how you called out OG Analytical, that like this industry has an opportunity to completely blow itself up or to collaborate with each other and give each other respect when respect is due. And I was like particularly touched when you did that. We could treat each other as competitors, we could treat each other with animosity and mistrust, or we could treat each other with respect and dignity and collaboration. So I'm hoping to bring that to this group. I think you all bring this to that group, to this group. And so email me if you have questions, go to the website, and then if you're not on the mailing list, Megan, will you raise your hand back there? She has a list. If you're somehow not on our mailing list, we will send out event notices through that mailing list, and we'll send out the Eventbrite notice to everyone who's on that list, whether you're a member or not. So thank you for coming. You guys are amazing, as always.